Hello, everyone, and welcome to today's symposium. I'm Charlene Lancaster, Assistant Science Editor for The Scientist, and I'll be moderating our discussion. Today, our expert panel will explore how scientists identify and characterize biomarkers for cancer, neurodegenerative diseases, and autoimmune disorders. Before we begin, I'd like to thank Fuji Rabio, Leica Microsystems, Selecta, Sampled, Ampersand Biosciences, Biorad, and Aviva for sponsoring today's webinar. We like our symposia to be interactive. We encourage you to send us your questions or comments at any point, and our panel will address these during the Q&A session following the presentations. To ask a question, simply click on the Q&A tab and type your query into the question box located on the right side of your screen. Our sponsors have provided us with some helpful resources related to today's webinar, and we've posted these in our handout section on the right side of your screen. You can access and download these documents at any time during the webinar. If you've minimized the Q&A panel, simply click on the blue chat button in the bottom right corner to bring it back. This symposium will be archived on the scientist website, and we will send you the link via email within a couple of days. Please note that you will not be able to download the presentation slides. And with that, let me introduce our first speaker. Jorge Moscat is the Homer T. Hearst III Professor of Oncology and Vice Chair for Cell and Cancer Pathobiology at Weill Cornell Medical College. He is also a Professor of Pathology and Cell and Developmental Biology. Moscat earned his PhD at the University of Madrid, focusing on signal transduction and hepatocytes, and completed postdoctoral training at the NIH, where he explored how RAS regulates lipid metabolism in cancer. His research focuses on kinases and autophagy modulators in tumor inflammation and has led to the discovery of novel signaling regulators. Let's make sure your slides are up and running.
Okay. Good morning, everyone.
Wait, I only see a blank screen.
Yeah, I don't know why. Allow screen share. No matter how many times you rehearse.
Always.
Oh boy. Okay, now I see. Okay. Do you see now my presentation?
Yes, we do. Okay, Jorge, take it away.
Okay, thank you very much for the invitation. It's a pleasure to be here sharing with you some very interesting data that we think we have on two proteins called P62 and MBR1 that have been proposed to be involved in the control of many signal transduction pathways and autophagy. And we believe they are critical regulators of hepatocellular carcinoma initiation and progression, and that could be true biomarkers to identify patients that could be susceptible to immune checkpoint blockade therapy. HCC, or hepatocellular carcinoma, is the second leading cause of cancer-related deaths in the world. It's a very quite heterogeneous disease, having identified more than 20,000 different somatic mutations that affect between 8 or 11 to 11 major signaling pathways and biological processes. But therefore, it makes the oncogene targeting therapy for this type of cancer extremely challenging. The evolution of HCC is very complex. It's HBV and HCV, viral infections are the current leading causes. However, thanks to the availability of vaccines, this is becoming much less prevalent, and obesity is rapidly growing and is important as an HCC risk factor. And I'll show you in a minute how that is so. It's a great relevance in the tumor microenvironment for the development of HCC, and this is also an important source to identify non-oncogenic specific vulnerabilities that allow us to really propose new treatments and identification of patients. Immunotherapy is a promising therapeutic strategy, but as you all know, is quite suboptimal, unfortunately. So that means that we require much more investigation on the mechanisms that leads to the disease in the point of view of the tumor microenvironment. Our laboratory has been interested for many years in this protein called p62 that is encoded by the, that is encoded by the gene sequestration 1. P62 has been implicated in our laboratory, in many other laboratories, in many functions, including the activation of mTOR, which is critical for sensing nutrients and controlling cell growth, thanks to interaction with a kinase like mechanism 3 or Raptor. And through mTOR, it blocks autophagy, but at the same time, P62, through the binding of LC3, a critical molecule in autophagy regulation, also promotes autophagy. So it's a critical regulator of homeostasis in autophagy pathways. You also have this domain called UBA, whereby it binds proteins that are polyubiquitinated and that target for destruction. So it's a critical regulator of the mechanism of quality control of proteins, mitochondria, for example. It binds TRAP6 and regulates NF-kappa B, so it's critical for inflammation. And more importantly, it binds this protein called KEAP1 that's an inhibitor of NERV2. By binding to KEAP1, sequestering KEAP1 away, activation of NERV2 really regulates the ability of cells to cope with oxidative stress, which is very, very important in hepatocellular carcinoma initiation and progression. We have published that P62 is a critical player in hepatocellular carcinoma, and the first evidence was when we're trying to understand how obesity really leads to HCC through two processes called MAS-LD and MASH. MAS-LD and MASH is metabolic dysfunction associated hepatitis, steatotic liver disease that initially was called NAFLD, non-alcoholic fatty liver disease, that progresses to MASH, which is called metabolic dysfunction associated steatohepatitis. And when people reach this stage, about 50% progress to cirrhosis, requires liver transplant, or unfortunately death, and 7% evolve to hepatocellular carcinoma. So it's clearly a disease that is very much depending on the microenvironment. We were interested in P62 initially because we found that P62 is highly upregulated in HCC progenitor cells. P62 is upregulated actually in all human chronic liver diseases that progress to HCC, and is a major component of the Mallory-Denk bodies that accumulate in human cirrhotic livers. We show that P62 not only correlates with the disease in human patients, but we also have cause and effect experiment in which we knock out P62 in the hepatocytes using albicree. So this is sequestration, the gene that flocks in albicree to selectively knock out P62 in hepatocytes. And we have developed experiments in 5 models. One is the DEN-HaFA diet that mimics the accumulation of transformation due to carcinogens with a progression promoted by obesity. The MAP-UPA model that was developed by Michael Kahn's laboratory of NASH, in which the mice recapitulate the whole progression from obesity to cirrhotic and hepatocellular carcinoma, the STAM model of the liver lesion, or the TSC2 knockout model that mimics overnutrition. In all 3 models, the loss of P62 really impair hepatocellular carcinoma development. In fact, the overexpression of P62, specifically in hepatocytes, also was sufficient to drive hepatocellular carcinoma initiation and progression in this case. So the mechanism is really bifunctional. On the one hand, P62 through mTOR activates proliferation of hepatocellular progenitor cells, and on the other hand, P62 through activation of NERV2 controls ROS levels in a way that ROS levels can induce mutation, however, cannot induce cell death because the levels are kept under more physiological conditions. So with this, that was very interesting because we thought that with P62 we can devise mechanisms that propose to revert MASH probably, and also we were interested in the role of the tumor microenvironment. The role of P62 in the tumor microenvironment, separated from its role in the hepatocytes, came from the analysis of expression of P62 in normal and tumor tissue from mice and human, which in a minute, in which we saw the normal hepatocytes express very low levels of P62, that are dramatically upregulated upon cell transformation. You can see these condensates of p62 that are signaling hubs to induce cancer. However, what we found as well in the normal part is that p62 was not expressed much in the hepatocytes, but was expressed in these cells over here that are also stained for desmin, alpha-SMA, which are hepatic stellate cells. Hepatic stellate cells are the precursor of cancer-associated fibroblasts when cancer happens, and myofibroblast. So p62 was really expressed in normal conditions by hepatic stellate cells at higher levels than it was in the hepatocytes themselves. However, when tumors happens, p62 is upregulated in hepatocytes and is downregulated in the hepatic stellate cells. You can see here that the hepatic stellate cell that we stain with desmin, it does not stand for p62, whereas the hepatocytes upregulate p62 dramatically, which is not in the normal situation. This is also true in human patients. We have analyzed a relatively large cohort of patients. You can see that patients are as well in the normal part of the liver, alpha-SMA, in this case, is the staining for the cells. They colocalize with p62. And however, this is completely gone with the tumors, and we can show that we can recapitulate this in vitro by incubation of hepatic stellate cells in vitro with HepG2, which are hepatic cancer cells. So we have a situation in which p62 upregulation in the hepatocytes in the tumor epithelium promotes transformation. However, associated with this, what we found is that p62 is downregulated in the hepatic stellate cells and cancer-associated fibroblasts. So this reduction of p62 begs the question whether p62 reduction promotes or favors HCC transformation. So we did two experiments that within that were revealing the role of p62 in the stellate cells. We used this mice called GFP p62 knockout, in which we selectively knock out p62 using the GFP promoter, or this other mouse model called LRAT, that we obtained from both Swabe and Columbia University. And then we compared these two mouse models in which p62 was not affected in the hepatocyte, was selectively knocked out the hepatic stellate cells. We used the two models, DEM, which is a carcinogen, in combination with carbon tetrachloride to mimic fibrotic liver. That's one of the characteristics of hepatocellular carcinoma. What you can see here that in contrast to what we have found and published in the hepatocyte, what we see is that the loss of p62 in hepatic stellate cells promotes a dramatic upregulation of tumorigenesis, number of tumors, and the tumor diameter marker of hepatocellular carcinoma. And the same is true in the other model, in the LRAT-3 model. The phenotype is quite similar to human patients. They have bridging fibrosis, inflammation, activation of inflammatory signals, and activation of SMA as a marker of activated hepatic stellate cells. So this is important because then what it means is that hepatocellular carcinoma is actually promoted by true antagonistic effect of p62. On the one hand, activation of p62 in the hepatocytes, in the hepatoma cells, concomitant with a reduction of p62 in the cancer-associated fibroblasts. The mechanism is quite interesting. It's sort of unrelated to what we knew before about p62 and all these pathways here that I described briefly in the beginning of my presentation. It's related to another protein called MBR1 that is a partner of p62 because they share this PD-1 domain that had been proposed to regulate autophagy like p62. The p62 PD-1 domain has two sides, an acidic and a basic side. Through the basic side, it binds to the PD-1 domain of MBR1, which is quite unique. They only have an acidic side. So the question is, if p62 bind to MBR1, they have a similar structure. Is MBR1 relevant in HCC as p62 is? So to address this question, we did the following model. We took wild-type mice, p62 knockout mice, the questosome is the name of the gene again, MBR1 knockout mice, a double knockout. Treat the mice with DEM, which is a hepatocarcinogen, and 60% half a diet to mimic progression through obesity. You can see in the absence of p62, we have a tremendous activation of hepatocellular carcinomas, not so in the MBR1 knockout. However, what was really interesting is that the double knockout MBR1 p62 completely rescue the phenotype of the PCT2 knockout mice. We rescue not the tumor number, but we will rescue the bigger tumor number. We rescue hepatocellular carcinoma progression, and actually mean that that MBR1 is required for the loss of PCT2 promote tumorigenesis. The question is this due to the role of P62 and MBR1 in the tumor microenvironment, in other words, in hepatic stellate cells? Yes, it is. And we showed this experiment in which we take a wild type, P62 knockout, MBR1, or the double knockout mice again. We implanted syngenic hepatocellular carcinoma cell line that we developed in our lab, DAH-XD3. And then we determine whether these hepatic cells, these hepatoma cells, these hepatic HCC cells can induce tumorigenesis in the wild type, in the PCT2, MBR1, the double knockout. What we found is that, in fact, we can see HCCs in the wild type, much more increased HCCs we find in the PCT2 knockout. MBR1 doesn't do much, it even reverts the effect on the wild type, and more importantly, totally rescues the effect on PCT2. So this is due to the role of MBR1 in the microenvironment, because the DAH-XD3 cells, they are proficient for PCT2, they are proficient for MBR1. So it's due to the microenvironment. And actually, it's due to the role of MBR1 in the hepatic stellate cells. And we showed this experiment with the mice that are either wild type for MBR1, or we have knockout MBR1 selectively in the hepatic stellate cell fraction. We treat the mice with half a diet again to mimic obesity, and we implanted the HCC syngenic cells in the liver. And interestingly, we find that the ability of these HCC cells to induce HCC in the liver in vivo is completely reverted by the lack of MBR1 in the hepatic stellate cells. The same is true in a co-implantation experiment in which we prepare hepatic stellate cells that are either wild type, PCT2 knockout, MBR1 knockout, or double knockout. We co-injected these hepatic stellate cells along with the HCC cells into the liver of these mice, again treated with half a diet to make obesity. And then we see clearly here that hepatic stellate cells that are deficient in PCT2 promote HCC in this co-implantation model, whereas the loss of MBR1 inhibits the effect on the wild type liver and reverts the role of P62. So what we have here, we think, is that both P62 and MBR1 in hepatic stellate cells play a critical role in hepatocarcinogenesis. And the role is opposite. On the one hand, the loss of P62 in stellate cells promotes HCC, independent of its role in hepatocytes. On the other hand, the loss of MBR1 in the context of PCT2 loss by itself reduces hepatocarcinogenesis. This suggests, therefore, that this being created a synthetic dependency between PCT2 and MBR1 that we thought was very important to explore. When we did was to do omics, bulk RNA-seq, comparing PCT2 knockout hepatic stellate cells versus wild type. And what we found is that associated to the loss of PCT2, there was a tremendous reduction in all the interferon signaling pathways. When we do the same approach, but in this case knocking out MBR1 instead of PCT2, in the hepatic stellate cell, we see the opposite, which is a tremendous increase in interferon, and that increase in interferon happens even though we can knock out PCT2 in the presence of MBR1. So the double knockout PCT2, MBR1 really also induces more interferon. It suggests that therefore that there's this synthetic dependency that we have found between PCT2 and MBR1 for hepatocarcinogenesis can really ascribe to effects in the interferon pathway. So we decided to explore this in more detail, and then what we did was the following experiment. In the mice that we have treated with what's injected with HCC syngenic cells and treated with half a diet, and then we have co-implanted with hepatic stellate cell wild type PCT2, MBR1, or double knockouts, you can see we see a very nice upregulation for phospho-1 in the wild type situation that is associated with recruitment of CD8. However, when the hepatic stellate cells are knocked out for P62, phospho-1 is totally reduced. CD8 is gone, the tumors are bigger. In the case of MBR1, the upregulation for phospho-1 is dramatically high. Tumors are small, and however, they are very, very rich in CD8s. And then in the double knockout, we can totally rescue the phenotype of the p62 knockout, in which we bring back phospho-STAT1 and CD8s. This is also true if we use the same experiment approach. Instead of using a co-implantation, we use this selective p62, MBR1 knockout mice in hepatic stellate cells. Now, our model, therefore, is that hepatic stellate cells that are knockout for MBR1 through the interferon pathway might be programming CD8 T cells to prevent HCC and to kill HCC cells. So this is shown in this experiment in which we use, again, we co-implanted wild-type mice that have been treated with half a diet with these HCC cells, along with hepatic stellate cells that are knockout for P62 and MBR1, and as I showed you before, do not produce tumors. These mice were injected with anti-CD8 to remove all CD8s from the system, and then we found that actually when we remove CD8, these double knockout hepatic stellate cells that cannot promote tumorigenesis are really restore this very aggressive HCC phenotype in terms of the tumor, in terms of lung metastasis that I show here, and the upregulation of the HCC process. So what is the mechanism? So we're very interested in understanding the mechanism because we think that it's not only important to understand that this could be a potential biomarkers and a protein, but we also want to understand how this works. There are two main pathways to induce interferon. One is the double-stranded RNase, RIG-I, MDA, MAPS pathway, and the other one is the response to double-stranded DNAs at cGAS that produce this second messenger called cGAMP that activates STING and interferon. So we did a series of experiments in our hepatic stellate cells where we found that when we knock out STING, the name of the STING gene is STING-M173, so sorry for the confusion. We knock out with a small interfering RNA, the STING, we knock out MAPS, you can see that only the knockout of STING totally abolished phospho-STAT1, the marker of interferon pathway, but there's no effect on MAPS. So that indicates, and we did a lot of experiments to prove that this was the case, in vitro, in vivo, and then that indicates, therefore, that MBR1 and P62 may be playing critical roles in the regulation of interferon through the activation of the STING pathway. So to address this question, we did the following experiment. We took, again, our favorite model, our mouse model, half a diet to mimic obesity. We injected them with hepatic stellate cells, with HCC cells, hepatocarcinoma cells, syngeneic, along with hepatic stellate cells that were either P62 knockout that promote tumorigenesis, the double knockout P62 MBR1 that prevents, that rescues the high tumorigenic effect of losing P62 in hepatic stellate cells, or hepatic stellate cells in which we have knocked down STING. Interestingly, when we knock down STING in the context of the double knockout, we can rescue perfectly well the ability of this of the P62 knockout to induce higher levels of tumorigenesis, as you can see here, tumor volume, also lung metastasis, you name it, all the phenotypes. Interestingly, what the hepatic stellate cells are doing through STING is to educate the CD8 T cells. When we isolate CD8 T cells from mice that are either a P62 knockout, have been implanted with hepatic stellate cells that are P62 knockout, or double knockout in which you had knocked down the STING gene, where you can see that in the context of MBR1 knockout, you have a generation of interferon gamma by CD8 T cells, by CD4 T cells, expression of MHC2 markers, and even activation of PD-L1 due to chronic activation of interferon that is totally abolished by the lack of STING, as you can see here. And the same is true when you look at all kinds of interferon-driven cytokines like interferon gamma, CXCL9, 10, etc. So our model, therefore, is that P62 and MBR1 somehow in hepatic stellate cells regulate the ability of cGAS to stimulate STING and to produce interferon. This is, we can see in vivo when we start interrogating what is going on in these hepatic stellate cells. Interestingly, what we did was in our model, we implanted the HCCs in these mice using MBR1 GFP3 that again knocks out MBR1 in the hepatic stellate cells. We can see here that in the wild type situation, And when we stain for alpha SMA, that we are detecting hepatocellular cells, there is not a STING expression under wild-type situation in tumors that are being produced by HCC cells. In the knockout GFAP3, in which we have knockout MBR1 selectively in the hepatocellular cells, we see a dramatic upregulation of STING in the hepatocellular cells. And we also see a very interesting field effect whereby there is an upregulation of STING not only in the hepatocellular cells, but also in the surrounding hepatocytes that we will explore in the future. But we'll do a quantitation. You can see here that the main phenotype in the MBR1 knockout hepatocellular cells is the upregulation of STING levels, and therefore that could account for activation of interferon pathway. So we have p62, that the loss of p62 somehow affects STING, and you have downregulation of MBR1, you restore STING and you restore interferon, showing again that we have this syntactic dependency between p62 and MBR1 for HCC and for the interferon pathway. Now, what is the mechanism? The mechanism we find is very interesting. As you know, STING have to travel from the endoplasmic reticulum to the cis Golgi, to the trans Golgi, and to the lysosome. And then by having when MBR1 is sitting in the Golgi system, particularly in the cis Golgi, it ensembles a macromolecular complex, including the TBK1, which is a kinase that phosphorylates IR3, that is important for induction of interferon. What we found is that we follow the transit of STING from the endoplasmic reticulum to the Golgi to the lysosome system. But we can see here staining for the Golgi. You can see that STING upon 30 minutes upon stimulation with SIGAMP, which is the product of the CGAS pathway, activated STING, it is really recruited to the Golgi, and then 60 minutes, 90 minutes later, it is really leaves the Golgi. In the MBR1 knockout, what happens is just that STING remains retained in the Golgi, and therefore is signaling much more efficiently because it's not being transported out from the Golgi, that's where all these complexes assemble, whereas P62 knockout has no effect on that. We study in great detail all this transport from the plasmic reticulum, cis Golgi, trans Golgi, late endosomes, and lysosomes. I will show you here. I don't have time to go to these details. Sufficient to say that by having the MBR1 knockout hepatocellular cells, that STING accumulates in the cis Golgi and therefore dramatically activate the interferon pathway. So STING binding MBR1 has two choices. One is to go to lysosomal degradation through this pathway that I showed you, through late endosomes, RAP7, or what we found is that when it is stimulated by CGAM, STING is binding P62 instead of MBR1. You can see here, if we IP STING, we wash the blood for MBR1, you see a very nice interaction at basal levels. Then when CGAM is added to the cell culture, MBR1 is expelled, and then P62 comes in to the STING complex. So what happens then when cells get stimulated, hepatocellular cells get stimulated, two complexes form, P62 carries MBR1 away from the STING, and P62 binds the STING, and that's what we think is important for the interferon response, because two things happen. We have showed this in a number of experiments showing, mapping and so on and so forth. So two things happens is that MBR1 is diverting STING, not from going to the lysosome anymore, but then becoming a signaling complex. Concomitant with that, what happened is that MBR1 binds this protein TRIN32. TRIN32 is an A3 ubiquitin ligase. I don't want to try you through details, but what is known is that for STING to be active, it has to be ubiquitinated. So what we found in all these experiments that I showed you here is that P62, as I showed you before, removes MBR1 for the STING complex. That's two things. On the one hand, prevents MBR1 to go into the lysosome, and then it sticks longer in the cis Golgi where all the complexes assemble. On the other hand, prevents MBR1 from inhibiting TRIN32. TRIN32 is constantly blocked by MBR1, and then what happens is that these two complexes, P62 MBR1, just keeps away MBR1 from the complex with STING, and then P62 orchestrates the formation of this complex, whereby TRIN32 induces ubiquitination of STING, Then this brings TBK1, this kinase that phosphorylate IL-3, this two transcription factor that trigger the whole interferon cascade. So what happened, therefore, is that we have a situation whereby P62 and MBR1 in a way play antagonistic roles in the regulation of the homeostasis interferon pathway in hepatic stellate cells. This is very important because hepatic stellate cells regulates interferon pathway that regulates CD8, and CD8 is the orchestrator of antitumor response to the generation of interferon gamma and killing of tumor cells, either cell autonomously or by recruiting CD8 that are active. So all this we would think is a very, very important mechanistic understanding of how the tumor microenvironment is really being impacted by the loss of P62 and MBR1. In mouse model, the question is if this is important for human patients and could be considered biomarkers for the identification of patients that could be susceptible to immune checkpoint blockade therapy. So the last 2-3 slides are going to show you data that we think support the notion that P62 and MBR1 are important regulators of these pathways in human patients. We obtained from our collaboration with Dr. Louis Robert in the Mayo Clinic in Rochester, Minnesota, a series of patients, of specimens from patients that were either normal livers, MASH, or HCC. As you remember, MASH is an obligate intermediate to HCC development. And what we found is that doing multiplex imaging for alpha-SMA to stain for the activated hepatic stellate cells, P62, MBR1, or DAPI, to identify nucleus. What we found, I'm going to go into details in this immunofluorescence, but I'm going to show you the quantitation. It's very interesting that as soon as a normal cell, a normal hepatic stellate cells, is in a MASH environment, it really loses P62, and that remains lost in HCC. I don't mean loss of genetically, I mean down-regulation at the protein level. We still don't know the mechanism, but that's what's happening. And concomitant with the loss of P62, we have upregulation of MBR1. So two things happens. From normal to NASH, from NASH to HCC, P62 is reduced, and that's important because reduction of P62 impairs interferon pathway, and MBR1 is upregulated, which is important because MBR1 also upregulation also helps to downregulate the interferon pathway. So we extended our studies to a large cohort, 164 HCC patients that comprised 287 samples, and we did multiplex staining in this case to compare alpha-SMA, STING, and MBR1. Interestingly, what we found is that hepatic stellate cells in these patients that have a negative, low level for MBR1, they have high expression of STING, of SMA, as compared to hepatic stellate cells that are positive for MBR1, indicating that upregulation of MBR1 is really killing STING in these cells. Consistent with this, what we have found is that there is a multivariate analysis. There is a very nice correlation between having low levels of STING in hepatic stellate cells with poorly differentiated phenotype in HCC that correlates with a positive expression of MBR1 in hepatic stellate cells. So with this, I want to leave you with the idea that we believe we have identified two proteins, P62 and MBR1, that were proposed to be critical regulator of autophagy. We know that P62 MBR1, we are not so convinced it's important autophagy. P62 is important in the control of autophagy, pathways that really regulate the growth of epithelial cells like hepatocytes when they become hepatocellular carcinoma cells. But in addition to that, they play a major role also in the tumor microenvironment. And the role that they play is opposite. And the role that they play is that the loss of P62 in hepatic stellate cells, concomitant with the upregulation of MBR1 in these hepatic stellate cells, they cooperate to reduce STING function and levels, which drives a reduction in interferon immunosurveillance, promoting hepatocarcinogenesis. And we want to propose that identifying patients that have this phenotype are the ones that are going to have the highest aggressive phenotype and the one that probably would be the least responsive to immune checkpoint blockade therapy. And with that, I will just finish acknowledging the people who did this work. The work was done in my laboratory work on medicine, collaboration with my colleague Maria Diaz-Meco, with whom I have a long-time collaboration. Part of the studies were done in collaboration also with my colleague Michael Carey at the University of California, San Diego. The samples were provided by Luis Roberts in the Mayo Clinic, and the work was done by a number of investigators, Angeles Duran, as an assistant professor in my laboratory, Juan Linares, Antoine Ermit, and Sadaki Nishimura, that was the last one that was in the laboratory and that really did all this work on hepatitis stellate cells. And with that, I would like to thank you for your attention. I'm happy to answer any questions.
All right. Thank you for that presentation, Jorge. We have several questions from the audience, so let's get to them. Given the opposing roles of P62 in hepatocytes and hepatic stellate cells, how might this complexity affect therapeutic targeting strategies?
Yeah, so that's very complicated. Actually, I totally agree with this question because you have to combine two things. You get rid of P62 in the hepatocytes, as we have shown, you prevent HCC. If you knock out P62 in the microenvironment, then particularly in the hepatic stellate cells, you promote HCC. And unfortunately, the one that wins is actually the loss of P62, because the total body P62 knockout mice, which are viable, they have the same phenotype that the hepatic stellate cells knockout. However, the critical question here is that we can really modulate this pathway, and we can modulate the response by devising ways to knock out MBR1. Because what happened concomitant to the loss of, so we have upregulation of P62 in the hepatocytes, loss of P62 in hepatic stellate cells, so the tumor is progressing because interferon is reduced, because the hepatic stellate cells, MBR1 is upregulated, that helps to get rid of STING. However, what we show in total body mice and in all the implantation experiments I show you, that if we knock out MBR1, we can totally restore the phenotype. If we knock out MBR1, it's a way to really restore interferon pathway and then potentially make these mice or these human patients hopefully susceptible to immune checkpoint blockade therapy. So even though P62 is complex and it's antagonistic in itself, the antagonism of MBR1 and hepatic stellate cells can be, I think, profit because it creates a vulnerability in these tumors.
Wonderful. And what are your next steps with this project?
So there are several things that we want to explore. Obviously, something that is in our mind is to how we can get rid of MBR1 with all this fancy technology that we have right now, these Lebron's possibilities, how we can really get tumors with MBR1 from knockout pharmacologically. This is one of the things we want to do. And the other thing we want to explore is how this pathway is placed in another major unmet clinical need. Part of my lab is very interested in colorectal cancer. And as you probably know, unmet clinical need is how to deal with patients that have colorectal cancer, that have formed liver metastasis. It is clear, it's obvious that obesity and the microenvironment plays a major role, not only in hepatocellular carcinoma, but also in metastasis progression and initiation in colorectal cancer, pancreatic cancer. So we have this other line of investigation that we think that we can really modulate MBR1 levels in this situation. We can also address the problem of metastasis from colorectal cancer and pancreatic cancer patients.
Excellent. Thank you, Jorge.
Thank you.
If anyone has further questions, please consider reaching out to the speaker directly. His contact is shown on the screen. So now let me introduce our next speaker. Katie Irwin is an MD PhD candidate in the neuroscience program at Johns Hopkins University School of Medicine. She earned her bachelor's degree from the University of Georgia and is currently conducting research under Philip Wong. Her work focuses on TDP-43 loss of function and neurodegenerative diseases such as ALS and Alzheimer's disease. Katie recently developed a novel biomarker that detects TDP-43 dysfunction at presymptomatic stages, advancing early diagnostics for ALS-FTD. So let's make sure your slides are up and running.
All right, thanks so much. I'll share my screen. Okay, is that looking okay for everyone?
It looks perfect. Thank you, Kay. So go ahead.
All right, thanks so much for the introduction and for the opportunity to present today on some of our work in Phil Wong's lab at Hopkins, as mentioned. So we've been working to develop a new fluid biomarker for TDP-43 loss of splicing repression in ALS-FTD. Oh no, okay. So as a lot of people are aware, a really major theme in different neurodegenerative diseases is the misfolding and aggregation of proteins. So in Alzheimer's disease, you see beta amyloid and tau aggregates. In Parkinson's, you see alpha-synuclein. In Huntington's, you see aggregates of the Huntington protein. And there are many more examples. But here today, we're going to focus on this protein called TDP-43, which is the main pathological hallmark of ALS, or amyotrophic lateral sclerosis. So ALS is a disorder of motor neurons. So both upper motor neurons and lower motor neurons degenerate in this disease, and it's a very quickly progressing disease with a typical life expectancy of only 2 to 5 years after diagnosis. And this disease, ALS, is actually linked to another disease called FTD, or frontotemporal dementia. So this is a disease that often presents with either behavioral or language abnormalities in this type of dementia. So these diseases are actually linked into a spectrum. So clinically, a lot of patients who develop ALS end up developing some symptoms of FTD, and also vice versa. A lot of patients who begin presenting with FTD will eventually develop motor symptoms. So these diseases are linked clinically, and they're also linked pathologically by this TDP-43 protein. So 97% of ALS cases involve TDP-43 proteinopathy, while around 45% of cases of FTD involve TDP-43 proteinopathy. So not only are they clinically and pathologically linked, but they're also genetically linked as well. So there are a lot of different mutations that can lead to either ALS or FTD, or a combination of the two diseases. So a big one that we'll end up talking about here today is the C9orf72 mutation. So this is a repeat expansion that occurs in both ALS, FTD, and often leads to both diseases at once. So what happens in these TDP-43 proteinopathies is you get these aggregates in the brain, in neurons of the TDP-43 protein that has been mislocalized from the nucleus and clumps up in the cytoplasm. So this TDP-43 protein is a DNA RNA binding protein, and it's involved in a lot of different functions in the cell. It's normally localized to the nucleus, like I mentioned, but it can shuttle back and forth between the nucleus and the cytoplasm. And it's involved in a lot of different things, a lot of which surround RNA regulation. And so because TDP-43 is in multiple compartments in the cell and is involved in so many different things, so many different functions across the cell, there's still a lot of debate as to what exactly related to TDP-43 is contributing to disease progression. So TDP-43, as I mentioned, is normally localized to the nucleus. So there's some theories that when TDP-43 is cleared from the nucleus in disease and aggregates in the cytoplasm, you could get loss of function effects from TDP-43 not being in the nucleus anymore. But also because it forms these pathological aggregates in the cytoplasm, there are theories surrounding potential toxic gain of function of this aggregated TDP-43. And this is all still very poorly understood in the field right now. However, we are interested in the nuclear clearance of TDP-43 as opposed to its cytoplasmic aggregation, because this is an earlier event than the actual frank cytoplasmic aggregation of the TDP-43 protein. So before you get these frank cytoplasmic aggregates of TDP-43 in the neurons, you're getting its loss from the nucleus. And we've actually seen that in patient tissues. So there's this one really interesting case in the literature where a woman underwent resection of her temporal lobe for treatment of a seizure disorder. And then actually 5 years after the resection of her temporal lobe, she developed symptoms of frontotemporal dementia. And so researchers were able to go back and look at her resected brain tissue from the presymptomatic stage of her disease. And they found neurons that lacked nuclear TDP-43, you can see here, but didn't yet have any cytoplasmic aggregates. So upon looking at her brain tissue, they didn't see any cytoplasmic aggregates of TDP-43 at this stage before symptoms developed. But then when this patient passed away, they were able to look at her autopsy tissues, and now at this end stage of disease, they did see these pathological cytoplasmic aggregates of the TDP-43 protein, in addition to the nuclear clearance, of course. So why is this nuclear clearance of TDP-43 important? We know it's an earlier event than the cytoplasmic aggregation in disease, but what could possibly be happening when TDP-43 is being cleared from the nucleus? So in 2015, this mechanism was actually discovered by our lab, whereby TDP-43 is actually really important in RNA splicing, particularly in repressing splicing of these non-conserved sequences called cryptic exons. So these are intronic sequences that are usually repressed from getting incorporated into the final RNA transcript by TDP-43. Specifically, it binds to these UG repeats that flank these cryptic exon regions in the intron and represses the splicing. However, in disease, TDP-43 is not in the nucleus anymore, so it can't serve the splicing repressor role. So these cryptic exons end up getting incorporated into the RNA transcripts, and then most of them end up introducing premature termination codons. So this leads to nonsense-mediated decay of the RNA. And this happens for the vast majority of cryptic exons that are inserted into RNA. So you can imagine that it could be problematic that with all these RNA transcripts getting degraded, you're losing all of those downstream proteins, many of which are probably very important for the cell. And so after this initial finding of the splicing repressor function of TDP-43, there were a lot of studies done into looking for particular cryptic exons and particularly disease-relevant ones that might have evidence for mechanistic consequences of this TDP-43 loss of splicing repression. So one major one that the field has focused on is this target called STAthmin-2. So it's a microtubule regulator, and it has been found to include a TDP-43-dependent cryptic exon. So when TDP-43 is lost from the nucleus of cells, a premature polyadenylation site actually gets incorporated into the STAthmin-2 RNA, which leads to an unstable truncated protein that's non-functional. So it's been shown that when STAthmin-2 is lost in these cells, there are impairments in basically neuronal response to injury, neurite outgrowth, and axonal regeneration are impaired. And this truncated STAthmin-2 target has actually been found in patient tissues as well. And then a second interesting target is in a gene called UNC13A. So UNC13A was actually previously shown to have SNPs that increased the risk of developing ALS and FTD. And then it was eventually found that there is actually a TDP-43-dependent cryptic exon in this UNC13A gene. And not only is there a cryptic exon in this UNC13A gene, but it's been shown that actually the risk-associated SNPs in this UNC13A gene, what those SNPs do is actually increase the incorporation of the TDP-43-dependent cryptic exon into the RNA transcript. So this evidence actually directly links genetic risk for ALS-FTD and TDP-43-dependent cryptic splicing. And these cryptic exons have been shown in patient brain. So this is not just something that's happening in cell models, but actually in human disease patient brains, you can see the incorporation of cryptic exons. These are just two cryptic exons of interest here. And these are specifically incorporated in the brains that have the TDP-43 pathology. So this is indeed happening in human disease, not just in cell models. And this, however, has only been shown in end-stage disease tissues. So currently there are several limitations in understanding this, this mechanism. So in ALS-FTD, we currently don't have any ways to actually probe TDP-43 proteinopathy or specific mechanistic consequences like loss of TDP-43 splicing repression in living patients. We're currently totally restricted to looking at postmortem tissues. So we're really only looking at end-stage disease, and that limits our understanding of what's going on during the disease process before a patient ends up passing away. So our lack of TDP-43 specific biomarkers has one, mechanistic consequences. or consequences for our understanding of the mechanistic aspect of the ALS-FTD disease spectrum. So while we have seen cryptic exons in patient tissues at end-stage disease, it's currently unclear whether this mechanism happens early in disease. And showing that it does happen early in disease would support the idea that perhaps this mechanism does have disease-driving consequences. And this would inform our understanding of the disease and our approach to therapeutic developments for the disease. And then additionally, this has clinical implications. So without biomarkers right now, diagnosis of ALS is often very delayed because there are a lot of disorders that present very similarly with motor symptoms, and it's very hard to distinguish ALS from these other diseases. And with already such a short disease time frame of just 2 to 5 years after diagnosis, any time that's lost to a delayed diagnosis is very significant. This also affects our ability to measure target engagement in clinical trials. If we don't have measurements where we can actually evaluate the output of TDP-43 function, it's hard to evaluate in clinical trials if we're actually restoring TDP-43 function. So our lab sought to solve this issue by harnessing the fact that while most of the cryptic exons that are incorporated from TDP-43 dysfunction are included out of frame in the transcript, as I previously mentioned, a small portion of these are actually inserted in frame such that they would be expected to be translated into these novel cryptic exon encoded peptide containing proteins. So because they're in frame, one would expect the rest of the protein to be intact, but there's this extra cryptic sequence that has been incorporated into the protein. So our lab thought that we could develop antibodies against these cryptic exon-encoded peptides and use them to actually detect these peptides in patient fluids as biomarkers of TDP-43 loss of splicing repression to answer these really important questions of, one, is this occurring really early in disease? And then two, can we actually turn this into a biomarker that we can use in patients to facilitate disease diagnosis? So first, we wanted to identify some potential targets of interest that might be good for biomarker development. So we had 3 major criteria for this. The first was, of course, that the cryptic exon is incorporated in frame, because if it's not, we wouldn't expect it to be translated into a protein that then we can detect in fluids. So here you can just see an example of 5 different cryptic exons that are spliced in. So these higher peaks are the canonical RNA sequences that are incorporated into these genes. But when TDP-43 is knocked down, you see these extra sequences that end up being spliced into some transcripts that shouldn't be there and are not there in the control RNA. And then we also wanted to pick targets that are expressed in relevant tissues. So if we're trying to study ALS-FTD, we wouldn't want to look at a cryptic exon that is only included in a gene that is expressed only in the kidney, for instance. So we selected cryptic exons in genes that are either expressed ubiquitously for relevance to all sorts of TDP-43 related diseases, or that are at least expressed in the central nervous system for relevance to our disease of interest here, the ALS-FTD disease spectrum. So you can see here, one example is this HGGFL2 gene. This is expressed ubiquitously, whereas there's another target of interest called ACTL6B, which is neuron specific. So this one would only be found in neurons. And then we also wanted to look at targets that would be expected to be immunogenic. So we used AlphaFold protein structure prediction software to actually model what the protein structure would look like with this cryptic peptide incorporated. So you can see here in this protein called HGGFL2 that the cryptic peptide is actually inserted and forms an alpha helix, which is a particularly immunogenic structure. So it would be predicted that antibodies might bind particularly well to this cryptic peptide. And this HGGFL2 protein is the target that I'm going to talk about here today. It's the main target we've developed so far. And this, as I mentioned a couple of slides ago, is a ubiquitously expressed target. So it could have relevance to all sorts of TDP-related diseases. So what we did with this target is we developed monoclonal antibodies against it. So we used a hybridoma approach to do so. And then we validated our different antibodies against this target and ended up coming up with one one top candidate to pursue into future assays. So how we screened the targets is we developed TDP43 knockdown cell lysates. So we used siRNA against TDP43 to knock down TDP43 in these cells compared to wild-type HeLa cells. And then you can see that in these TDP knockdown HeLa cells, the RNA transcript corresponding to the cryptic HTGFL2 exon is included in the TDP knockdown cells, but not on the control cells. And then an antibody against the wild type, the native HTGFL2 sequence, detects a band of the expected size in both the TDP knockdown and the control lysate, while our novel cryptic HTGFL2 antibody detects a band of the expected size only in the TDP knockdown lysate. And then when we use these antibodies in conjunction, so when we do an immunoprecipitation with the cryptic antibody and then probe with the wild type antibody on a blot, we get a band of the expected size, again, only in the TDP knockdown lysate and not in the control, suggesting that this combination of antibodies is specific to our target of interest. And these other bands here are just the immunoglobulin bands, by the way. So after developing an antibody that is specific for our target, we wanted to see if this cryptic HTGFL2 protein is even present in human disease before we go looking for it in the biofluids where it would be harder to detect. So we looked in postmortem brain tissue in the motor cortex and the hippocampus of ALS-FTD cases compared to controls. And here you can see that our cryptic HTG antibody shows that cryptic HTG accumulates in neurons, specifically those that have been depleted of nuclear TDP43. You can see these gaps in the TDP43 staining, and that also have, in these cases, cytoplasmic aggregates of the phosphorylated TDP43 protein. So this shows that in human disease, we can actually find this protein, and our antibody specifically detects it in the cells that one would expect it to be found in, those that are missing nuclear TDP43. So then, having seen that this protein does indeed exist in human disease, we developed a sandwich ELISA on the MSD platform to go ahead and see if we can detect this, this novel protein in patient biofluids. So we developed the sandwich ELISA where we capture with our antibody against the cryptic HTGFL2 protein, and then we detect with a sulfo-tagged antibody against the wild-type version of the of the protein. So first we screened this assay in just cell lysates. So we looked at cell lysates either overexpressing the cryptic HTGFL2 protein or the wild type HTGFL2 protein. And you can see that there's a concentration dependent increase in MSD signal for the cryptic lysate compared to the wild type lysate. And then we also wanted to ensure that this difference in signal is indeed specific to the antibodies that we're employing. So what we did is we ran the assay under normal conditions, seeing an increase in the cryptic signal compared to the wild type signal. We also ran it with a denatured capture antibody, which would be expected to not bind to the cryptic protein anymore, and then also with isotype control capture and detection antibodies. And you can see that when we switch out these antibodies with either non-functional or non-specific antibodies, we lose the increase in signal for the cryptic lysate compared to the wild-type lysate. So then, having established an assay that is sensitive and specific for cryptic HTG, we moved forward with testing CSF samples from disease. So here we looked at just a small pilot cohort of ALS CSF compared to control CSF, and you'll recall these patients have that genetic mutation that I was saying causes either ALS-FTD or frequently a combination of ALS-FTD. So this is familial. These are 5 familial ALS cases. You can see that in this pilot cohort, we saw an increase in the cryptic HTGFL2 signal compared to the control CSF. And then employing the same antibody controls that I showed you earlier, we see that this MSD signal coming from the ALS CSF is indeed specific to the intact and correct capture and detection antibodies. So then after this pilot, we moved forward with testing a much larger cohort. So we looked at almost 300 CSF samples here. And there's a lot of numbers here, so I'll try to walk you through it. So we expanded our cohort into a group of controls, both younger, healthy controls, and then older controls, where one might actually expect to see a few cases of elevated cryptic HCG because TDP43 dysfunction, aging is a risk factor for that. So we had both younger and older controls, and then we looked at both sporadic ALS and familial C9orf72 linked ALS. This is important because looking at a genetic mutation cohort allows us to actually look presymptomatically as well, because this is basically a 100% rate, this genetic mutation, of developing ALS-FTD or both. So we can actually look at presymptomatic individuals who are going to develop disease at some point and see if in that presymptomatic stage, we can already detect elevated levels of cryptic HCG. And indeed, in this cohort, we could. So elevated levels of cryptic HCG were found in all of our disease groups, including the presymptomatic C9 mutation carrier group compared to controls. And not only were the levels higher, but the percentage of people in the cohort that had positive signal was higher as well. So then we wanted to explore how this actually might relate to different disease variables. So we found this interesting negative relationship between cryptic HCG levels in ALS-FTD and disease duration, suggesting that cryptic HCG levels tended to be higher earlier in disease. And so we looked at neurofilament levels in these CSF samples as well, because neurofilament is really the only other biomarker that is currently being studied or employed in, or mostly employed in ALS-FTD. And neurofilament is an intermediate filament that is found in neurons, and it tends to be elevated even greater in ALS than in other neurologically relevant diseases, because in ALS, it's really these large motor axons that are degenerating. So you get a really massive release of neurofilaments into CSF. And so we wanted to look at how our biomarker relates to neurofilament. And while our biomarker tended to be higher earlier in disease duration, we actually see that neurofilament levels didn't tend to be higher until about 2 years into disease. And this kind of corresponds with what other groups have seen. So others that have studied neurofilament in ALS have shown that neurofilament levels can increase in a few months or so before actual phenoconversion, where patients are diagnosed with ALS-FTD, and then levels tend to increase from there. So because we had only looked cross-sectionally, we wanted to see longitudinally how do our cryptic HCG levels compare to neurofilament levels over time, because potentially there might be a novel biomarker that's elevated even earlier and could facilitate diagnosis when used in conjunction with neurofilament, better than neurofilament alone. So fortunately in this cohort, we did have longitudinal samples, so we were able to look within an individual over time and see how cryptic HCG levels and how neurofilament levels changed. So each line here represents one individual with multiple CSF samplings across time. And you can see here that while pretty much no one had elevated neurofilament levels presymptomatically in this cohort, we already see several individuals who have elevated cryptic HCG levels in the presymptomatic stage. And not only are they already elevated, but in many of these individuals, the levels are already starting to decrease before these people have even developed symptoms. And then in the symptomatic stage of C9orf72 linked disease, the cryptic HCG levels are decreasing over time in all of the individuals shown here. Whereas in contrast, you can see that in neurofilament, in the early stages of symptomatic disease, the neurofilament levels are increasing, and they don't decrease until later in symptomatic disease. So using this longitudinal data, we put together this proposed model of these different biomarker trajectories over time, where we see that in some individuals, cryptic HCG can actually be elevated and even peak in the presymptomatic stage of disease, and then tends to decrease over time. Whereas in contrast, neurofilament can be elevated a bit presymptomatically, but this peak tends to occur later than cryptic HCG-FL2. So because of these differential trajectories, we thought that perhaps we could take a ratio of the neurofilament levels over the cryptic HCG levels, and this could provide a staging scale of presymptomatic to symptomatic disease. So we did calculate these measurements for our CSF samples in this cohort, and you can see here that all of the presymptomatic individuals have a neurofilament to HCG ratio less than 1, as expected based on these curve dynamics. And then most of our symptomatic carriers have neurofilament to HCG ratio is greater than 1, again, as predicted by these dynamics that we see here in the symptomatic stage of disease. So all of this work was in CSF and was very promising and exciting for us that we can see this biomarker presymptomatically, suggesting that this is an early mechanism in disease and one that can be harnessed for potentially designing therapeutic approaches and also for facilitating earlier diagnosis in the future. But of course, acquiring spinal fluid is not as ideal as acquiring blood. Blood is much more clinically useful and much easier for patients to donate. So we wanted to see if we could detect this biomarker in blood as well as in CSF. So some of this cohort, we had matching blood samples corresponding to the same time points of collection as our CSF samples. And you could see that in many C9orf72 mutation carriers, we did see elevated levels of cryptic HCG in the blood. And these levels were actually significantly correlated with those in the CSF. So this biomarker is detectable in blood in addition to CSF. And going forward, we hope to further optimize this assay so that we can expand the clinical utility of this biomarker target. So in summary, cryptic HCG is detectable in CSF of sporadic ALS and CSF and blood of presymptomatic and symptomatic C9 mutation carriers. So this means that TDP43 loss of splicing repression is not just an end-stage phenomenon, but it's actually involved in early-stage disease and even pre-symptomatic stage of disease, which supports the idea that this potentially does have disease-driving consequences for disease. And additionally, when looking longitudinally, we found that cryptic HCG levels tend to decrease over the course of symptomatic disease and may even peak pre-symptomatically. And this trend is offset from that of neurofilament, suggesting that when used in conjunction, these two biomarkers could be even stronger used together than individually. Additionally, evaluating the dynamics of cryptic peptide biomarkers could provide a way of measuring target engagement for therapeutics developed in the future to actually restore the function of TDP43 splicing repression. And then finally, this evidence suggests that identifying either nuclear depletion of TDP43 or TDP43-dependent cryptic peptides in postmortem tissues and histological sections might provide a more sensitive approach to classification of TDP43 pathology, because currently the standard of confirming whether a patient does have TDP43 pathology or not is looking for these cytoplasmic aggregates in cells. And now we're seeing that patients can have effects of TDP43 misregulation in a stage of disease where there are actually no cytoplasmic aggregates yet. So potentially we're missing a lot of cases upon autopsy that do have TDP43 dysfunction. They just don't yet have frank cytoplasmic aggregates. So potentially expanding the way we look at TDP43 pathology in postmortem tissues could help us better understand the disease as well. So I'd like to thank the Phil Wong Lab, all of our members, as well as all of our collaborators, especially at the biorepositories that provided us samples for this study, and of course the patients themselves who were involved in these studies to donate samples. And I'd be happy to take questions if there's time.
And we have a few questions to start off. How might these findings affect diagnostic practices for ALS and FTD?
Yeah, so I mean, obviously it'll take some time to actually get this to the clinic. But what we're hoping is that because our assay is working in blood now, and we have some newer data that with a newly optimized version of the blood assay that's working quite well, hopefully that expands not only the utility of this biomarker for familial disease, where patients have a known genetic mutation that points them to the fact that, you know, at some point they will develop this disease. This biomarker can be useful for them for hopefully predicting symptom onset in those individuals. But because we've expanded to blood, hopefully it can be useful in sporadic disease as well, because while if you don't have any symptoms, you're not going to get a lumbar puncture just to see what might be going on, but you will get blood drawn. So potentially this could even be used presymptomatically for those with sporadic disease as well. Of course, though, there are multiple TDP43 related diseases though. So it will be important going forward in the future to develop a panel of additional cryptic targets, as well as looping in other biomarkers that are known to be indicative of certain diseases, to create a panel that helps us really in the pre-symptomatic stage of disease, not only just point to risk of developing TDP-43 related disease, but actually which one. So hopefully as we develop additional cryptic peptide biomarkers, different cryptic peptide signatures in conjunction with other biomarkers can actually point to a certain disease pathway that a patient might be on. So that's where we see this going, hopefully in the future.
And is that where your lab is currently heading towards, looking at other cryptic proteins potentially?
Yes. So we're currently, I briefly mentioned another target called ACTL6B. That's a neuron-specific target. We have an assay working for that right now that we're working on gathering more cohorts to study. But that one we're really interested in because it is neuron-specific, whereas HGGFL2 is ubiquitous. So we can combine those two. It actually will give us more information on where specifically the cryptic peptide is coming from, because right now we can't confirm it's actually from neurons, right? That's the assumption because those are the cells that are being affected in these diseases. But if we can actually measure that in conjunction with a neuron-specific peptide and see if those levels either change similarly or if there are differences, that'll give us more information about disease. So we're working on that, on developing other cryptic targets, and then on developing a multiplex panel where we can lump in our cryptic targets with other well-established biomarkers.
Wonderful. And what causes the migration of TDP-43 out of the nucleus?
Yeah, that's a great question. We as a field do not know. There's a lot of studies trying to decipher that. There might be multiple upstream pathways that can misregulate TDP-43 and lead to its clearance from the nucleus. But right now, that's a major question in the field that people are working to unveil.
Perfect. All right. Well, thank you, Katie. If anyone has further questions, please consider reaching out to the speaker directly. Her contact is shown on the screen. So next we have Francesca Di Simone, a scientific affairs manager at Fuji-Rabio Diagnostics, Inc. She leads the company's neurodegenerative disease program, where she develops clinical evidence for biomarker portfolios and evaluates new technologies for diagnostics. Francesca holds a PhD and completed her postdoctoral fellowship in neuroscience and neurovirology at Temple University. So let's make sure our slides are up and running. Okay, Francesca, take it away. Oh, you're still muted.
Can you hear me now?
Yeah, I can hear you now.
Wonderful. Thank you so much. And again, apologies for that. I will reshare my screen. Hopefully it won't take me a minute. Okay, can you see my screen again?
Yeah.
Wonderful. Thank you so much. Again, good morning, everybody. It's a pleasure to be here, and I'm really happy to have the opportunity to kind of give this presentation and really kind of giving a little bit of a history, right? Describing the evolution of AD diagnosis and really kind of trying to focus on the important role the biomarkers have played over the years and how really, again, that technology has developed, especially, again, if we're considering the latest and greatest, again, in this field. So as we know, again, Alzheimer's disease is the most common cause of dementia, and it does account for an estimated 60% up to 80% of cases. And AD is characterized by the buildup of proteins in the form of amyloid plaques and neurofibrillary tangles in the brain. And according to the Alzheimer's Association, in 2025, approximately 7.2 million Americans aged 65 and older are currently living with Alzheimer's dementia. And also, according to the Alzheimer's Association, this number is projected to then nearly double, reaching 14 million by 2060. And so, of course, with the increased number of prevalence, again, of AD is, of course, closely tied to the rising cost of dementia care. So once again, Alzheimer's Association indicates that in 2025, the health and long-term care costs for people with dementia are expected to reach $384 billion. And this projects the costs are supposed to hit the $1 trillion by 2050. So really kind of emphasizing this huge social and economical impact of this disease. So as I mentioned earlier, again, my goal was really to kind of give an overview of the evolution of the clinical aspect of AD, how the biology evolved through time, but also kind of hinting at those clinical diagnosis guidelines that have basically provided more information and more guidance, again, how to navigate the disease, but also how to navigate and how to utilize correctly biomarkers. And so again, as you can see within this timeline, we have made great progress, I guess, over the past few years. So even if we already start looking at, 2011, when the NIAA came out with the recommendation indicating that AD biomarkers were grouped into two specific groups, so the A and the T-related neurodegeneration, which, of course, was per se like a very important step, because again, it was kind of already giving a better understanding of the disease and also kind of starting hinting at that framework with the ATN, but really again kind of had some limitations and wasn't really addressing the full-blown picture when it comes again to Alzheimer's. Soon after, we see, again, the FDA approval of the first amyloid PET tracer, so florbetapir, so really kind of sticking up to the idea that biomarkers are now picking up the pace. And then in 2018, the NIAA came out with a revised criteria, which really kind of shifted the gears a little bit and changed the perspective. So really now we're moving into a biological definition of Alzheimer's, which really indicates that cognitively impairment is now looked as a sign and symptom rather than the actual disease itself. So moving across our timeline, of course, 2021 was an incredibly exciting year with the FDA accelerated approval of the first disease-modifying therapy, so aducanumab. And then soon after, in 2022, the first FDA-authorized CSF beta-amyloid ratio on LomoPulse, once again, aimed to assess amyloid in patients with early AD dementia. And then, of course, 2023, 2024, the approval of two new DMTs, so Leqembi and Kinsula. And then I'm sure that you are aware, a few days ago, again, the FDA cleared the first plasma biomarker, so the LomoPulse p-tau217 over A beta 42 ratio, once again aiming to assess amyloidosis in the context of early AD. And then last but not least, I also think it's incredibly important and relevant to the field, the fact that we're really months away from the first publication, again, of the blood-based biomarker guideline for clinical practice and clinical use from the Alzheimer's Association. So really kind of indicating that, everything is moving so fast, but now we're actually starting to have some recommendations and guidelines that can kind of help us through the process. So as I mentioned earlier, we know that currently we have two FDA-approved DMTs, but of course, again, we know that there are a lot of pharma companies that are really kind of working hard in working on their drug development pipeline. So Cummings in 2024, came out with this really beautiful overview paper that kind of addressed different aspects, but really also highlighted a few important points. So, for example, for the disease-modifying therapies, it actually emphasized the importance of early detection of the disease, precisely because these drugs are known to be most effective in the early stages within the continuum. Additionally, most phase 3 clinical trials do require evidence of amyloid positivity for trial inclusion, which indeed basically requires confirmation with different approaches, which could be, again, the CSF measurement, PET imaging, but also opening up the door to plasma evaluations. And then last but not least, they also indicated the important role that biomarkers can play in the drug development program. So again, not only as trial entry criterion, but also as measurement, allowing the measurement for secondary outcome, but also for target engagement. So speaking about, obviously, the guideline or the recommendation, in 2024, the NIAA came out with a new revised criteria, which I think really emphasized, again, the shift and the change that has happened, again, over the past year or so. So really the implementation of blood-based biomarkers in, again, the context of AD, and we'll talk a little bit about this in a few slides, but also giving more value and more granularity, again, to the actual biomarker category that you should be looking at. So really kind of expanding that ATN framework that has been, referred to over the past years, and really kind of trying to add more nuance and more flavor to that, to really kind of appreciate each and every stage within the continuum. And so specifically, again, with the core 1 biomarkers, we have the amyloids for A, so again, for the amyloid proteinopathy. And of course, we have the implementation of amyloid beta 42 species, potentially in combinations, such as ratios as well. And then with the readout with imaging through amyloid PET. But interestingly, again, within the 2024 NIAA revised criteria, we now have core 1 T1 biomarkers, which are really related to the phosphorylated forms of tau. So specifically the p-tau 217, p-tau 181, and p-tau 231. And these really represent an important class of biomarkers, because although they are part, again, of the tau species, or the phosphorylated tau species that should, again, reflect more tau pathology, they're actually really good at picking up those earlier stages within the continuum, and therefore they're basically playing a dual role, allowing also for really early detection of the amyloid in the amyloid stage. And then, of course, moving into the core 2 T2, which again is a full-blown tau pathology. So we have biomarkers such as MTBR tau 243 or phospho-tau 205, and then as a readout with imaging through tau PET. Additionally, again, we know that AD is a multifactorial pathology, and so we really do need to understand more about additional pathways that may be adding more fuel, again, to the process, and basically are pushing the patient through the continuum. And so, of course, that includes, again, markers for neurodegeneration, so obviously NFL being the key biomarker of interest, but also opening up to inflammation with as a readout of astrocytic activation, so of course GFAP, but also understanding the importance of non-AD co-pathology biomarkers such as vascular brain injury and alpha-synuclein. So why do we need biomarkers to support amyloid and, to some extent, also tau assessment? So currently we know that between 25 and 30% of patients within a clinical with a clinical diagnosis of AD are currently misdiagnosed, even in specialty dementia clinics. And this rate of misdiagnosis goes up to 50% in primary care clinics. And so this really kind of highlights and emphasizes the fact that diagnosis in early stages, so really kind of looking at that MCI stage, but also moving a little bit earlier where patients are already experiencing some symptoms that may not be picked up as well with biomarkers. Assessing those patients and identifying, again, the risk of developing AD down the line is really very difficult. But ironically, that's really the population that you want to target in order to have these therapeutics being the most effective. So as I mentioned, again, historically, the first biomarker that has been introduced was indeed amyloid PET. And so for the past 20 years, amyloid PET imaging has allowed visualization and quantification of insoluble amyloid protein fibrillary deposit. So really kind of allowing that total definition of the total load and spatial distribution of the amyloid beta plaques. So indeed, it does present incredible, positive features in the sense that, again, it does allow early identification of patients destined to develop AD and really kind of picking up these patients years before the actual onset of dementia, and really kind of enhancing the understanding of the pathology itself and also the related neurodegenerative disorders that are indeed associated with cognitive impairment. So currently, again, there are 3 FDA approved PET tracers, so really kind of the fluorine 18 labeled PET tracers, so florbetapir, flutemetamol, and florbetaben, that again have been approved for the assessment of amyloid pathology within the US, thanks to the FDA, but also in the European Medicine Agency. And in general, again, the published sensitivity of these tracers range between 82 and 98%, whereas specificities range between 77 and 95%. So moving into more recent years, again, there's been a big rush in trying to kind of move a little bit away from PET imaging and try to kind of evaluate additional approaches. So of course, CSF has become, of great interest, and because it really kind of, it's incredibly informative, and it does provide you with a relatively non-invasive look into ongoing pathological changes that are actually occurring in the brain. So of course, it can really represent an important tool, not only in the research field, but also in clinical practice. And that's pretty much because, as we know, the CSF serves as this mechanical and immunological protection for the brain. And therefore, again, a direct measurement of these biomarkers within the brain, within the CSF, allows you to have a perfect snapshot of where we are in terms of biochemical changes. And so when we talk about CSF biomarkers in the context of early amyloid detection or early amyloidosis, we're really kind of always discussing or really looking at A beta 42 and potential combinations of that, which has shown, again, to have great concordance with plaque burden at autopsy, as well as great concordance with amyloid PET. And this has also been indicated in some clinical trials from the EMERGE and ENGAGE studies that really reported this high concordance with both amyloid CSF and amyloid PET, so really kind of reaching that AUC of 90%. And that's, of course, against in the context of amyloid PET with the visual read, which is the FDA-approved methodology. So really kind of this indicates that CSF is a reliable alternative to amyloid PET. But what's actually interesting that has been established now, through multiple studies, is that there are some cases where the patient may receive both CSF measurements and PET imaging, but turns out that the patient does have a discordant result where the CSF is positive and the PET read is actually negative. So what that really means is that potentially the CSF, the amyloid species that it's in the CSF is actually being picked up a little bit earlier, and therefore it kind of provides you that additional layer of advantage, if you may, because again, you're kind of pushing the boundaries and you're moving a little bit earlier on in the continuum. And again, currently we have 3 FDA-authorized assays for the Fujirebio Lomopos amyloid ratio. That again was the first biomarker to be authorized, and then of course the Roche Alexis p-tau 181/42 ratio and the total tau 181/42 ratio. So, what are the pros and cons, so the limitations for PET and CSF? So, coming back to PET, again, we know that it's a very well-established approach. As I just mentioned, again, there are 3 FDA-approved tracers, and PET is in general very well tolerated, and there are very few contraindications. But there are some limitations to this approach. Of course, it's a very expensive tool, technique. So specifically, we're looking at roughly $6,000 at the imaging center compared to a cost of $18,000 at the hospital setting. It does require, specialized personnel. So, and it's also, it kind of also is its availability is a little bit impacted. So again, it's very limited geographically, if you may. And it only provides you with this dichotomous read. So it's either positive or negative. So you kind of may missing some of the nuances there. On the other hand, CSF biomarkers are indeed less expensive than PET, have a high concordance with PET, as I just showed. And in general, lumbar puncture is safe and well tolerated. And then last but not least, insurance typically do reimburse most of the cost of the CSF biomarkers. However, again, this approach also presents some limitations. So although lumbar puncture is safe and well tolerated, it's still perceived as an invasive tool. And there are some contraindication, again, for lumbar puncture. So for example, patients that are on anticoagulants should not undergo lumbar puncture. And of course, there are also a few side effects that do come from this procedure, including headaches and back pain, that kind of really, play a negative role on how the patient is perceiving this technique. And so in general, again, when we discuss, CSF biomarker measurements or imaging in general, the problems there are that they are invasive, they are time consuming, they can be very expensive, and again, in most cases, they're also very the availability of these, for example, PET scans is very limited. So once again, what is the option? So over the past year, blood-based biomarkers, again, have really held great promise to revolutionize the diagnostic and prognostic workup of AD in clinical practice. And so really kind of highlighting those advantages over CSF and PET. So of course, we know that, they're not invasive, they're perceived as safe, and really kind of blood draws are routinely used worldwide. They're highly scalable. There is therefore easy accessibility. And of course, there is also this important cost approach. So again, they're very cheap compared to the previous two approaches. And then last but not least, it may also allow for retrospective blood analysis in clinical research or clinical trials. Obviously, this with the caveat that samples do have to be stored in a proper way and handled in a proper way. So kind of really following those guidelines in order to reduce, any pre-analytical mishandling that could then lead to potential misdiagnosis. So again, kind of wrapping up on how we progressed and where we are now, over the past year, again, there's been a lot of effort trying to kind of really understand how do we position this blood-based biomarker? What is the greatest clinical benefit? So one of the main approaches that has been suggested is indeed the triage approach. And that is available thanks to the implementation of these dual cutoffs. So this high cutoff and low cutoff that kind of helps distribute the patient into 3 categories. And so specifically, the patient that is experiencing cognitive symptoms receives a blood draw. And then if the test result is positive, that kind of confirms that the symptoms of the patient is experiencing is in line with amyloid pathology. And therefore, again, it kind of confirmed that it is AD. And of course, the patient can then potentially access anti-A-beta treatments. On the flip side, again, if the patient receives a negative test result, it kind of rules out the possibility of this patient having amyloid pathology associated with AD, and therefore it kind of redirects the patient to additional care or alternative care. Now, the patients that kind of fall within these dual cutoffs kind of have a indeterminate or intermediate result, which basically is indicating that the patient was not, could not be classified as positive, could not be classified as negative, therefore requires an additional confirmation via either amyloid CSF or PET scan. And so really the goal here is to, have the CSF and PET scan there as a confirmation test, but with the goal really to kind of reduce to a minimum the patient that would receive this intermediate test result. And of course, then streamline the patient to appropriate care. So additionally, last year, the COI also presented a recommendation from the BBM work group, which really provided a wonderful guideline to clinicians and experts in the field to really understand what is the acceptable performance of blood-based biomarkers. And so it really kind of gave some indication in the context of, again, prevalence. So if we are looking at, for example, specialty cares where we're expecting high prevalence of disease, what the performance of the assay should be or is required to be. And then, of course, that's also implemented for the low prevalence. So again, kind of in the 20% area, where again, it's kind of reflecting the primary care setting with a low prevalence of disease. So again, it kind of really gave a nice rule looking at sensitivity and specificity requirements for each of these categories, and also, again, factoring in the prevalence. So kind of having this readout with the PPV and NPV as well. But most interestingly, it also provided some requirement, once again, in terms of sensitivity and specificity, in order to understand if an assay may actually be implemented as a triage test that we just described earlier, or it can actually have the potential to be used as a confirmation test. So really kind of streamlining the importance of reaching that high sensitivity and specificity of 90%. So really, as a reminder, the goal here is to have a test that is highly sensitive in order to avoid false negative results, but also have an assay that is capable of high specificity in order to avoid the excessive false positive result, while reducing to the minimum, again, the population that may then require confirmation. So trying to kind of looking at that percentage as less than 20%. So which once again brings me now to the first IVD test that, again, has been cleared again by the FDA last Friday. And indeed, again, this really represents a major breakthrough in Alzheimer's disease because it really enables earlier intervention and it does improve access to these disease-modifying therapies. So really with the ultimate goal of enhancing the quality of life of both the patient and the caregiver as well. So indeed, the future looks bright now because we do have the availabilities of DMTs, and we're now finally streamlining again the access again to either therapeutics or even again to appropriate care. And so last but not least, with integration of blood-based biomarkers into the patient journey, again, there are some different nuances on how we can position the blood-based biomarkers I we just mentioned, but also what can we look at for the future. So again, the current potential uses of blood-based biomarkers, which have been validated in a good cohort, indeed do provide this dual result, where again, in case of a negative test result, the patient, the provider will then identify other causes for these symptoms and therefore proceed to appropriate non-AD care. But then on the flip side of the coin, if the result is positive, then that will actually allow the patient to potentially initiate treatment or symptomatic treatment when appropriate. But what about the future? So looking at potential uses of blood-based biomarkers, of course, one of the most important and interesting aspects would be to be able to kind of screen those healthy individuals that are at high risk of developing AD. But of course, in order to approach that, we will need a big study just to kind of really address that very important question. But then, of course, blood-based biomarkers do present a very potential role also in tracking disease progression, so throughout the continuum, but also treatment response, so really kind of helping those patients and really kind of helping the physician also to kind of come up with a more personalized approach for each patient. So with that, again, I thank you so much for your attention, and I'm looking forward to any questions that you may have for me. Thank you.
Wonderful. Thank you, Francesca. So let's get to the questions. Which biomarker is most effective for diagnosing Alzheimer's disease, considering the required high concordance with cerebrospinal fluid, amyloid ratio, and amyloid positron emission tomography?
That's a very great question that kind of brings me a little bit back to, again, the ATM framework, right? So one thing that we do need to, take into consideration is when we talk about biomarkers in the context of PET or CSF, and in some cases also, again, plasma now, we do need to understand that we're kind of looking at the amyloid assessment within the ATM framework. And so once again, when we do consider the ATM framework and all the, the recommendations that have been provided, we have seen, again, extraordinary performance of the plasma p-tau217 species, as I mentioned earlier, which is kind of falling into that core 1 T1 group. So really kind of being able to pick up those early nuances, but also, again, coming back to the very recent clearance, the potential implementation of ratios. And that really is the idea behind, so the idea behind that approach is that we do know, again, that 217 can be a very early biomarker, but p-tau217 does have quite a dynamic ability per se. So the implementation of an amyloid species may actually help looking at the dynamics of the two protein, pick up earlier changes, and therefore kind of really pushing down, I'm really trying to achieve, again, the goal of early detection.
Great. And are there blood-based biomarkers expected to completely replace the CSF and PET scans?
Wonderful question. So that, I think the best way to approach that is kind of chiming back in with the COI guideline. And so indeed, again, once we implement that 2 cutoff model, we're kind of looking at this stratification of the patient with a positive, negative, or an intermediate result, right? But in that context, again, with the COI guideline, that is already kind of setting the stage on what are the expectations. So again, that kind of sets the stage for the triaging. So yes, we could have positive, negative, and then a group of patients that would then require additional confirmation with CSF or PET. But once we move into those higher performing assays, so again, kind of hitting those 90% sensitivity and specificity, the recommendation now is kind of shifting slightly, and it's kind of suggesting more blood-based biomarkers as confirmation tests. So again, I think, there's a lot of work being done. I think that we are nearly there. And again, the FDA clearance kind of is hinting at the fact that, yes, we do have very valid assays out there that can reach those performance. So I think, again, we are on the right, road for that one.
That's great. And are there any key limitations and concerns kind of moving forward with integrating these biomarkers into clinical practice?
Absolutely. So I think some of the key, concerns can include, for example, variability between assay platforms or even the lack of standardization. So it's true that, again, there is a lot of effort currently in the field to kind of fill those gaps. And so there is a work, again, trying to standardize this assay. And indeed, also, again, the need of regulatory approval. Once again, now we do have, again, one biomarker that has been cleared. So all of these things kind of have been factored into as potential key limitations. And then, of course, there is the ethical consideration around the early diagnosis and the potential stigma. So all of this kind of need to be considered very thoroughly in order to kind of really, have full trust, if you may, again, of blood-based biomarkers. But once again, I think based on the data that we're seeing and the studies and, again, the efforts and the guidelines, I think, again, we're kind of narrowing those perplexities, if you may.
That's great news. And how might blood-based biomarkers influence accessibility and equity in Alzheimer's disease diagnosis across various healthcare settings?
Sure. So that's kind of coming back a little bit to the discussion that I touched on with the PET, for example, PET accessibility. So again, there are some areas, including rural areas or under-resourced clinics, where, again, accessibility to a PET scan or even, having the personnel that can actually, perform a lumbar puncture may be problematic. So the goal here, and I think that's really kind of also the excitement around blood-based biomarkers, is that the implementation of blood-based biomarkers will significantly help reduce the geographic and socioeconomic disparities in diagnosis and care. And so, again, really kind of allowing earlier accessibility to everybody and hopefully, again, access to drugs in order to kind of help patients, but also caregivers, which is always the end goal for all of us that are working hard in this field.
Well, thank you, Francesca. If anyone has any further questions, please consider reaching out to the speaker directly. Her contact is shown on the screen. So unfortunately, David Nathanson is unable to join us today, so we'll move on to our next speaker. Luis Alvarez studied physical chemistry at University of Paris-Sud, where he specialized in fluorescence lifetime imaging. He has worked on host pathogen interactions at the University College Dublin, the National Children's Research Centre, and later at the University of Oxford, where he studied viral entry mechanisms of HIV and Ebola. At Leica Microsystems, he is currently a product application manager developing an advanced imaging techniques in fluorescence lifetime, live nanoscopy, and high multiplexing. Let's make sure our slides are up and running.
Give me just 1 second.
Of course.
You should now see my presentation.
Yes, we can see it. Perfect. Go ahead and take it away.
Great. Thank you so much, Harleen. And I'm going to switch gears a little bit. Thank you for that presentation on blood-based biomarkers. And I think we're going to look a little bit at a different perspective of how biomarkers are important and fundamental for understanding disease. I will share today an example on high multiplexing imaging in cancer immunology, but these approaches actually applied for a broad understanding of different pathologies. And I want to start with something that, spatial biology has become a buzzword lately, but the reality is that we've been doing spatial biology for a long time, and we're trying to describe biology through markers. At the end, all of these markers are the essence of how we can understand the hallmarks of what we see and what we observe, since the first description of the cell, or what Ramon and Cajal was showing us, and all of the histopathology we have seen today. Now, historically, all of these observations are done in very thin samples of 5 microns to 10 microns thick, and this is because of the technologies that we have used at the end to try to understand, to have a little bit of a meaning and topology of what's going on in vivo. Yet, it is a little bit challenging to push this a little bit further. And as we have been looking at this from our perspective, we started questioning, okay, if we have a given biomarker or a given mechanism, what is the likelihood that we will find exactly what we want to in these really thin samples? And so we thought that actually bringing together all of the new technology we've been developing in the field with biomarkers, but also enhancing the ability to look at this further in 3 dimensions, could actually bring us closer to understanding the mechanistic relationships between what's going on in vivo. So in the example I'm going to show you, we're essentially having this mouse model of KBC pancreatic tumor that we're putting into the C57 black 6 mice. And what we're going to be doing is actually harvesting the tumor and doing 70 micron sections. And then the idea is to actually try to understand the relationship between what's going on with the different markers here. I have put here the reference if you want to have a little bit more information on the specific methods to get this going on. And this is work that we have done in collaboration with um, uh, 2 researchers from Roche in Switzerland. And let's look at this a little bit closer. The main thing here is that if you're just looking at one set of markers, like the immune markers, well, you're going to be missing part of the story. You will be able to tell where the immune cells are, but you might not be able to see actually the effect that they will have on the cancer. And for these, we will have some functional markers that will actually tell us a little bit the state of the cells and how they're affecting the tissue. In the same sense, we would need actually additional markers to be able to have proper context. And I think when we look at some of the images, it will become clear how actually bringing all of these 3 kinds of markers together can really enhance our view and get us closer to understanding what's going on. This is important as a descriptive method, and actually a lot of the high multiplexing up to now are very descriptive, trying to characterize what is in a tissue. But most importantly is actually having this subset and having the 3 dimensionality will allow us actually to go further and do mechanistic studies. So as you can see, this is quite rich in for all of you that are used to doing fluorescence. You might not be used to packing all of these uh, fluorophores together, but what we have behind it in our system is a way to have very clear excitation. So we basically freely excite in the whole visible spectrum, and we can do a separation as that is very well defined. Again, in the links, you will be able to have more details, but you can see that if we were looking at cells, actually, for us, it has become routine to actually follow 4 different markers directly without any problem. And this is the basis to actually go chase the high multiplexing and now in 3D. Now, this is very important because there are certain limitations. So most people have been using the iterative approaches, but we decided to do a one shot because this will avoid actually first the destaining and the staining rounds do damage the tissue, and they limit actually the accessibility of your markers. So this is one of the reasons why most of these approaches are really limited to 5 microns, 8 microns if you're pushing it. But anytime you go beyond that, actually, you have really big problems with the penetration of a lot of the chemicals that you need to do to destain and restain. Now, this gives us an additional valuable asset, and is to preserve this tissue. I mean, we are excising tissue and we're cutting it, which means that actually you always have small damage, but having such large tissue sections at 70 microns or more means that you can be sure that you have significant portions of the 3D structures and the 3D relationships that are preserved. And to have all of these then actually will allow us to really look at all of these objects and how they work together. So let's have a look a little bit. So this is 3 by 2.5 millimeter tissue section in which we have put these 50 markers on a go. I will just play this movie so that you can see actually more in detail how every part of this tissue is very rich, and it allows you to actually start asking the questions of what is the relationship between the immune cells, their effect in tumorigenesis, and how the context, the tissue context actually is going to affect this. Here are some regions of interest that are 550 by 550 microns in width, in XY, and still the 70 microns thick section. And one thing that for me is striking is that when I take these regions of interest individually, it would be difficult to know that this is coming from the exact same tumor tissue. And you can appreciate, I hope, the very difficulty you would have to be able to actually catch all of these relationships if we were doing just a very thin section of tissue, or if we only were limited to a few markers. So just like the work that we have seen this morning on cancer, and also on Alzheimer's, actually, once you have found a biomarker and you can actually see the implications and you can see that there's an effect either in mice or in humans, then actually being able to go back and put this biomarker in context can really allow you to start probing what are the functions, what are the other parts that are integral to triggering the disease that might actually help you find alternatives for intervention, or trying to look at other synergistic markers that you could have. You can see here that actually you have a really nice topology of not only the lymph cells, but also the TCF1 and PD1 expression, where shows actually what are the areas in which is really affecting the tumor, and how actually all of the structure of the tumor has been organized around this. Now, just I want to touch briefly on the technicalities on how we can get there. So this is actually an approach that we call SpectraPlex, that in this case we have shown with 50 markers, but this is not a limitation, we can do more. And it turns around trying to change the perspective of doing imaging from just putting a bunch of colors to actually concentrating on what defines a biomarker, meaning the panel. So looking at the targets and actually pairing these so that you can keep track of what are you imaging. So not necessarily, the fluorophore, the DAPI or this, but actually what is your target and how this is going to give you the answers that you want. One of the things that is very important is your controls, as the results and the fidelity of the results is only as good as your scientific controls. And I'll touch a word or two on this side. And finally, to be able to just acquire all of the data without having to worry about the quality of the data, because all of the pipeline ensures that you have a faithful representation of the tissue and what you have. Now, just a quick word here. So working with a lot of biomarkers at once is really information rich, but it comes from some challenges. And the challenges is that having so many signals together, you need to ensure that all of the data is pristine and that you have very little uncertainty of where the signals are coming from. For this, actually, you need to validate actually your controls. And historically, what people have had to do is do single strain controls, which, yes, is the gold standard. But you can imagine that if you're looking at human tissue, I work a lot on host pathogen, and we were studying HIV infections and Ebola, I can tell you that whenever we had human samples, the last thing we could afford is to have a multiple of controls just to be able to run one experiment. With that in mind, we have actually developed a way in which you can have the same fidelity of controls, grouping them together in a way that is guided through SpectraPlex, and it tells you exactly what are the parts that you can put together to have exactly the same quality of data. This intelligent guidance actually then selects this and allows you to see exactly what is the source of all of the fluorophores, and to be able to have the mixing matrices that you will need to be able to actually get to your results later on. So again, one of the things, and is a little bit philosophical that we did, is you can see here the acquired tab is actually locked until you do your controls. We feel that actually this is very important, as we have heard in the previous talks. Actually, there's a lot of conclusions that come from these kind of data sets, looking at biomarkers. What is the perspective? What is the effect on the cancer? What is the effect on certain drugs? And you want to ensure that all of the results that you're getting out are as high quality as possible. So again, what do we do? Actually, we can actually have all of our markers staying in one go on our tissue. And from there, actually just get the 3D 15-plex confocal image. Because we have a full confocal there, actually the thickness is not a problem, and we can have all of the details that we need in all of the markers that we have. So this is on-the-fly algorithms allows you to actually see directly after acquisition the results without having to wait, and be able to actually assess the quality of the sample and of the data directly. All of this optical sectioning and the resolution also allows you to have a high fidelity of where the signals are coming from. And if you see the interaction of two cell types, you can be sure that this is indeed the case. And if you need to further image with higher resolution or such, you can go back and do so. Now, this is only part of the question of biomarkers. Once you have such rich information, then what's next? And what's next is that you need tools that allow you to generate insights. And this is actually, we're very excited, is very developing field. And as part of that, we have developed an Aviva workflow. This is our AI generated software in which we can have now the analysis of 15 plus plex 3D images, and allows you to do first the segmentation, but then the classification, so that you can really map out the interactions and be able to actually get all of the information that you need to be able to actually see which are the biomarkers that are coming together or that are correlated or anti-correlated if needed. Here's an example of the 15-plex from adenocarcinoma mouse tissue section, and how Avia can detect all 15-plex cells. And from this, actually, we can start building the classifications and the relationships that will then allow you to really understand how much of the biomarker is involved with which cells at what state, and what is the fate of all of the processes together in the tissue towards one thing. We can see that we can look at the co-expression of the functional markers, and then from there, the interesting thing is that the biomarker is no longer a yes or no answer. Is it present or not? But also, what is the spatial relationship between them? As we can see, the fact that a given biomarker is present is already indicative very often of a disease and disease progression. But understanding the spatial relationship with all of the other players can actually allow you to really assess the fate of, in this case, tumorigenesis, but actually of most pathologies, as the development of disease is quite complex and is very seldom just a linear progression into our direction. So these tools allow you to have user-guided but unsupervised phenotyping of all of the tissue with all of the biomarkers, and be able to then actually explore things that you might otherwise miss because you're only looking at one or two biomarkers without the context. Here we can see in this latter type that we can identify really nicely the immunosuppressive phenotype compared to the helper T cells. And actually, from this characterization, we can go back into the image and actually find the areas that have this and see if there is particular other signals that we might not have been aware of. Again, all of these tools allows you to really have a complete landscape and a complete overview of what's really going on in your tissue, and explore further what are the implications in drug discovery, and also if you're looking at inhibitors or just disease progression on its own. So I want to just finish with a recapitulation of what SpectraPlex for Stellaris is, what it can do. A big part of this you can find on an application note we published in Nature Method in October last year, in which we have the experimental design for advanced exploration of multiple markers in 3D. Here we have actually the flexibility that allows you to use almost any fluorophore present, because we have all of the visible spectra that is accessible both in the excitation and the emission path. The full confocal capabilities that allow you to really do true 3D and be able to go beyond just a thin section in wide field with low resolution. And the integration to all of these platforms with the analysis pipelines and the ability to, if you already have your own pipeline, to be able to do the segmentations and actually have links to other open source resources such as Cytomap and others. Here's a QR code that you can use to read this Nature Methods application note. And we have actually published in November a second application note that actually has all of the mathematics and all of the mixing algorithms that allow you to have high fidelity to mapping out all of these biomarkers in 3D using this reduced amount of samples as controls that we think is really important to allow you to do high multiplexing even in rare samples that you would not dare to just waste in doing 15, 20 or more controls at once. So I want to finish by just thanking the people that have made this possible. So we have a very strong Leica team, in particular with Dr. Susanna Holzmeister, the R&D project lead, and Dr. Julia Roberti, the product manager, and our collaborators in Roche that work with us on the October Nature Methods, and the collaborators at MIT Harvard from the lab from Professor Harry Wong that actually collaborated with us and published together with us this application in the November Nature Methods. So thank you very much for your attention, and I'm happy to take any questions.
Thank you, Luis. So let's dive into the audience questions. So first off, spectral flow cytometry is becoming more and more popular. What are the advantages of using spectral imaging compared to flow cytometry?
So actually, this is very complementary. Now, flow cytometry spectrally is almost the norm in many immunological labs. I would say 16 colors is very routine. And the difference is to do flow cytometry, you essentially have to divide all of the cells, and you will only have the percentage of content per cells. In the imaging, on top of this, you can actually see what is the relationship between the different targets that you have identified. One thing that is of interest is that usually if you show up to an imaging center with flow cytometry antibodies, they will look at you and just say, please change all of that, and give you a set of fluorophores. But the flexibility that we have on the Stellaris platform actually has allowed us to use many of the flow cytometry antibodies as part of our panels. So if you're doing flow cytometry spectrally already, and you would be interested in seeing how the spatial relationships affect your results, you could actually just take your tissue with the exact same panel and give a SpectraPlex a go. I mean, it might not work 100%, but it's an extremely good starting point, and you might need very little adaptation, actually.
So if there was someone that was interested in getting into multiplexing imaging, what advice would you give them?
Read a lot. No, it's an interesting. So there's a lot of. First, I would suggest define your biological question very well. So there's a lot of imaging platforms out there, and a lot of them have very restrictive or focus, if you may say, questions that they can answer. They're very seldom very flexible, and you may want to double check that whatever your question can be answered with that imaging approach. I am very biased because I was part of the team that developed SpectraPlex, but one of the things that for us was capital in SpectraPlex is the flexibility to ensure that it's not only one thing. So I have shown right now some results from cancer that I think are really, really nice and innovative. But we also, so I'm based in Germany, and we have some collaborators at the EMBL working on single molecular RNA, for example, 10, 12 plexes. We have people working on skin as well with high panels with very different topics that are not restrictive to just cancer. So imaging now is evolving rapidly, and make sure you formulate your question and make sure that whatever imaging approach you're going to use allows you to answer that. And again, have a look at SpectraPlex. It's very likely that you can image with the solution we just came out with.
Wonderful. Well, thank you, Luis. If anyone has any further questions, please consider reaching out to the speaker directly. His contact is shown on the screen. Now let me introduce our final speaker. Lisa Willis is an assistant professor in the Department of Biological Sciences at the University of Alberta and adjunct professor in medical microbiology and immunology. She earned her PhD from the University of Guelph, completed a Banting postdoctoral fellowship at the University of Toronto, and now researches immune sugars and their role in diagnostics for diseases like scleroderma. Her work spans biochemistry, glycoimmunology, and sex-based immune differences. So let's make sure your slides are up and running. Perfect.
Yeah,
All right, Lisa, take it away. Yeah.
Awesome. Thanks so much. And thank you, Louis, for introducing spectral flow cytometry. I'm going to talk a little bit about that. So what I wanted to do today was actually something a little bit different from what the previous speakers have done. They've been coming from a tissue and a disease and making their way towards a biomarker, and we've been coming at it from the opposite angle. And it all started with coffee. So this was just before COVID. I knew nothing about the autoimmune disease scleroderma. I happened to have written a grant application. We'd been doing a lot of work in cancer, and I'd written a grant application. We wanted to see if some of the things that we were doing might be applicable to other autoimmune or inflammatory diseases. And I'd written a grant about lupus. And I had someone at the university read it and say, no, lupus is the wrong indication. You need to talk to this person right here, Mo Osman, about the autoimmune disease, or rather autoinflammatory disease, scleroderma. And so this started with coffee, and it's grown to be a really spectacular collaboration with some of the people in my lab and some of the people in Mo's lab who have all done the work that I'm going to show you today. So the other thing I'm going to do is slightly different, is that I'm not going to tell you a complete story. I'm going to take you on the journey that has gotten us to where we are, and we are very much mid-story. We found some really cool things that we think are promising in diagnosing and prognosticating scleroderma, but it's actually going to leave you on a cliffhanger. So there will be a stay tuned at the end. The first thing I need to do is to introduce you to the to scleroderma. A lot of you, like me at the time, might not have heard of it because it's actually very rare, only about 40 per million, 40 to 80 per million, depending on where you are. It's an inflammatory disease that of the skin. It's characterized by fibrosis, and it starts off as a sort of a less severe form and can progress to a more severe form. So I'm showing this picture of Raynaud's phenomenon. Raynaud's phenomenon is actually really common. About 5% of the population has it, and it's where the ends of your fingers turn white when they're exposed to cold usually. So this is really, really common. But almost everyone who has scleroderma actually starts out with Raynaud's phenomenon. And what we're looking at here are the capillaries that are in the nail bed here. So this is nail fold video capillaroscopy. And what you're looking at here are the capillaries, and you can see that they're all nice and even. They're relatively narrow. They're uniform. And so this is what a normal capillary bed looks like. This is from someone with Raynaud's phenomenon. But then in scleroderma, you start to lose that uniformity. The capillaries become thicker. You start to see some microhemorrhages. Your fingers get puffier, and that makes it actually a little bit difficult to use your joints. This is an early pattern, and then you have an active pattern where you start to see lots of hemorrhages. Your joints become really thick. Your extremities become really thick, and you start to get thickening of the skin as well. And then you see a late pattern here where all of the normal capillary structure is gone. We don't actually know what causes scleroderma. There's some sort of epithelial cell injury and remodeling of the vasculature. There is an associated immune dysregulation, and it's not clear which one precipitates the disease or how they interact with one another. It leads to inflammation, and then eventually fibrosis, which is excessive production of these extracellular matrix components like collagen. We have no idea what causes the disease. It's linked to genotoxic and oxidative stress. People who work with jet fuel, for example, are more likely to get the disease. But we don't know what causes it. And there are no curative therapies. There aren't even really good treatments for this disease. The only curative therapy that we know of is autologous stem cell transplantation. This is an awful therapy where they essentially collect stem cells, then they treat you with really horrible chemotherapies to kill all of the fibrotic cells, and then they reconstitute those stem cells. It's only used for a few patients, and it's associated with severe complications. So 5 to 10% of people just die from the treatment itself, and then 30 to 40% relapse. So there are two major types of scleroderma. The first is called limited, and this is where the symptoms are mostly limited to your extremities. So that doesn't sound too bad, but truly this is one of the diseases that has a really high morbidity, and is really uncomfortable, it's painful. It really compromises quality of life, for sure. And then we have diffuse scleroderma. And this is where the fibrosis also occurs in the internal organs. And so you get fibrosis in the heart and lungs, and that leads to a really high mortality rate. This disease, the average age of diagnosis is between 30 and 50 years of age. So we're not talking about older adults. We are talking about young adults, people who have young families. It is far more common in women than in men. about 80 to 90% of people living with scleroderma are women. And the disease trajectory is actually quite different in women and in men. So in women, about 2/3 of people will remain with limited scleroderma. And only about 1/3 will progress to diffuse scleroderma. Whereas in men, even though it's far rarer for men to have the disease, most of them progress to diffuse scleroderma. And this is a problem for many reasons. One is that the autologous stem cell transplantation, that's only used in people who we know are going to end up with diffuse scleroderma. So in men, it is possible, because we know that that's where they're likely going to go, it's possible to actually treat with the stem cell transplantation earlier, before extensive tissue damage happens. And the earlier you treat, the better the prognosis is. But the same isn't true for women, because we don't know how the disease is going to progress in women. We can't predict. There's no way to predict. That means we have to wait until the diagnosis of diffuse scleroderma already exists. And by then, frequently, it's too late. This is a very quick, in a lot of people, it's a very rapid disease. I know of people who have gone from diagnosis to death within 2 months. So it can be very, very quick. And so we have a huge number of unmet clinical needs in scleroderma. The fact that it's a rare disease means sometimes it's difficult to get patient samples. It doesn't attract as much attention. There are fewer people working on it. So we really don't understand the mechanisms of the disease. There is no good animal model, so it's challenging to actually study the disease in the way that you've heard earlier today. And so that's left us with a huge number of questions. How do we predict who will respond to the stem cell transplantation? If we can predict who will respond, then we can focus efforts on those. How do we predict who is going to progress, especially in women? Because, like I said, in men, we know what's going to, it's far easier to tell what's going to happen. But how do we predict who will progress so that we can treat earlier? How do we prevent people from developing diffuse scleroderma and getting that fibrosis in their heart and lungs? How do we alleviate the symptoms in any type of scleroderma? Because even if you don't end up with diffuse, you live with really horrible symptoms. And then finally, sort of the pie in the sky, how are we going to cure scleroderma? And I don't have answers for any of the bottom three, but we are starting to address the top two. So this is where I came in. I am a basic scientist. I study how glycans control the immune system in health and disease. What you may not know is that every single cell on this planet is completely coated with a layer of sugars. And when you think of sugar, you might think of table sugar, glucose, sucrose, fructose, the things that you eat. But there are lots of different kinds of sugars. There are 9 in humans, and they form polymers that coat the surface of every single cell. So what we're looking at here, this is actually a blood vessel, and this is the inside of a blood vessel. Here we have the endothelial layer, and then all of this stuff here, this is glycans. This is all sugars. And I'm really interested in these because they're on the outside, they're on the surface of the cell. They're what's mediating the interaction between that cell and its environment. That is what the cell uses to sense its environment. And so I'm really interested in how these sugars mediate the immune response in health and disease. So the sugar that I'm going to tell you about is called polysialic acid, or polysia for short. The sugars that I'm talking about today are attached to proteins. So here's a cell membrane, here's a protein, and then there are sugars attached to it. And the ones that I'm interested in are the purple diamonds. And purple diamonds occur everywhere, but purple diamonds in a long chain, that's what polysialic acid is. And this is unique. So it's only found in a few different places in the human body: the nervous system, the immune system, and the reproductive system. That's the only place you would find this in a healthy human adult. And it mediates cell migration. So anytime you have new neurons forming, synaptogenesis, as they're coming together, those neurites are coming together, those cells are polysialylated. And then it also attenuates immune responses. So it's very involved in trimming of cells that shouldn't be there, in mediating the migration of immune cells to areas of the body where they need to go. And we're very interested in understanding more about the immune side of this glycan. But crucially, it can also become dysregulated in disease. And the one that's best studied is cancer. So it doesn't matter where in the body the cancer originates from. Cancer, any of these potential areas, can become polysialylated. So cells in the skin, for example, that would not normally express polysia, re-express them when they become a cancer cell. And if they do that, they are more likely, that cancer is more likely to be metastatic. So it's going to migrate to other areas of the body. And the presence of polysia strongly correlates with poor prognosis. And this is what we were studying. But it's also, polysia is dysregulated in mental health disorders, in neurodegeneration, and we've shown recently in inflammatory diseases. So one of the things I want here is our spectral flow cytometry. We had been looking at polysialic acid in the immune system, and we had been seeing that smokers actually have higher numbers of polysia positive cells. So just looking across the different major immune subtypes here, we've got polysia in every major immune cell type. And I'm sorry, I always forget that the legend is missing on this slide, but the open circles, these are non-smokers, and the closed gray circles, these are smokers. So polysia, there are higher numbers of polysialylated cells in monocytes, a little bit in B cells, and a little bit in T cells. There's a trend towards higher numbers. And we did a deeper dive into a couple of different immune cell types. I'm showing you T cells here. In the CD4 compartment, for those of you non-immunologists, these are the coaches standing on the side of the field yelling at the players. There's really no difference in numbers of polysia. But in the CD8 compartment, these are the players. There, you start to see higher polysia in smokers, especially as they become more differentiated. So especially in the T effector population, and the amount of polysia per cell is also higher. And we had also found that the effect was greater, especially in the CD8 T effector population, in males. So there are no sex differences in polysia in healthy males and females. They look identical. But in smokers, we started to see differences. So here's the no difference in non-smokers between females and males. We see an increase in females and smokers, and also an increase in males who are smokers, but the increase is bigger in magnitude in males. And so it's significantly different in males. And then looking at the amount of polysia per cell, we see no differences in non-smokers. We also see no difference in the female smokers, but the male smokers have higher levels of polysia per cell. So we'd been seeing sex differences in immune polysia. We'd also been looking at cancer and seeing that cells that shouldn't normally be making polysia are making polysia. And so there are actually a few similarities between scleroderma and cancer. It's almost like a cancer-adjacent inflammatory disease. You see the same kind of differences in metabolism. You get Warburg metabolism. You see DNA damage. You see oxidative stress. All of these kinds of things that you would see in a cancer, you also see in scleroderma. And so then we asked the very simple question, very naive question, of whether or not polysia might actually be dysregulated in skin of patients with scleroderma, Are there also sex differences, and could it be used as a biomarker? So you see what I mean about coming at this from a very different angle. We were not studying scleroderma. We were not looking at the differences between healthy skin and scleroderma skin. We came at it from this really bold, novel, kind of crazy approach of, hey, is this a thing? And it turned out it was a thing. So I don't know if this is a plug for this kind of science, but this is the kind of stuff that really excites me. Is let's just try it and see what happens. So what we're looking at here are sections of skin from healthy controls, patients with the less severe form of the disease, limited scleroderma, and patients with diffuse scleroderma. And what you can see here is that, and it's quantified over here, what you can see is that polycia levels are much higher in the skin of patients with scleroderma, and it correlates with disease severity. We were really interested to see that post-autologous stem cell transplantation, those polysialic acid levels really go down. And this is, so you would only get this transplant if you were originally a diffuse scleroderma patient. And so this difference is also very interesting to us. Again, we're looking in humans right now, so it's difficult to really fully get at causation, but we have some really nice correlations here. We wanted to know if polycia was actually coming from the fibroblasts. So in scleroderma, the cell type that's mediating that fibrosis are the fibroblasts. That's what's secreting the fibronectin and the collagen. And so we looked at patient-derived fibroblasts. We take biopsies, we culture the fibroblasts and isolate the fibroblasts, and then analyze them. In healthy controls, as you would expect, this is an immunoblot looking for polycia. In healthy controls, as expected, there's no polycia. In patients with the limited form of the disease, you can see polycia, and then in patients with diffuse scleroderma, you see more polycia. And what's interesting to us, this is not the best comparison, but these don't look similar to us. They look different. It looks to me like there are 2 or 3 proteins here, and this is obviously much, much more intense in signal, but it looks more like one protein. So we think there might be some differences between the early limited and the diffuse scleroderma. We looked at the expression level of the 2 enzymes that could be responsible for making polysialic acid, so ST8-SIA2 and ST8-SIA4. We found that both of them are significantly upregulated in patients with scleroderma, and again, correlate with disease progression. So this kind of mirrored what we were seeing with cancer, in that cells that should not normally express polycia start to re-express it. And we had this hypothesis in cancer that some of these cells, some of these proteins, rather, these glycoproteins, were actually being secreted and could maybe be used as a biomarker. So we're going to move to what we were doing in cancer for a second. We had developed this assay where we could take a 96-well plate, we could coat it with a protein that binds to the glycan. We could then put in serum or whatever complex mixture we wanted. That would bind to the glycan and whatever protein it was attached to. And then we could come in with an antibody either to the glycan itself or to a specific protein and use that in an ELISA assay to detect whether the glycan, quantify the glycan or proteins that it was attached to. So in the context of breast cancer, I'm showing you the quantification of polysialic acid in healthy controls. These are all women. In situ, ductal carcinoma in situ. These 2 here are in people who had breast cancer. So one of the things that was interesting was that the amount of polycia that we saw didn't actually correlate with the stage or the grade at time of diagnosis. All of these samples are serum taken at time of diagnosis. And for us, the levels did not correlate with the stage or the grade. What it correlated more with was the outcome. So in patients who survived to 5 years, that's what we're looking at here. And in patients for whom the disease was fatal within 5 years, that's what we're looking at here. So we saw a trend. It's nothing to write home about, but it's definitely a trend. And we weren't sure if the proteins here were coming from the skin itself, or in this case, the breast cancer itself. Was it coming from those cells, or was it coming from a dysregulated immune system? And so we looked at the protein that is most commonly polysilated in the immune system. There were no differences here. And so we reasoned that it was probably coming from the breast cancer itself. And so we wanted to determine, could we actually figure out what the proteins were and maybe develop a better assay? Now, normally the way you would do this would be to immunoprecipitate the polysilated proteins. You would take an antibody to polysia, you would mix it with your serum, you would isolate that antibody, and then you would do proteomics on anything that came out of that. The problem is with polysialic acid that a lot of the proteins they're attached to are membrane proteins, which makes them difficult to isolate sometimes. The glycan itself is large and negatively charged, and so it tends to bind to other things that are that bind to large polyanions, like ribosomal proteins and histone proteins. And so immunoprecipitation can be quite challenging with this particular system. This is data from a different lab where they identified neuropilin-2 as a novel polysilated protein. But you can see from this data, they only found one peptide with neuropilin-2. It's not exactly compelling. And there were lots of other proteins in here. It turned out that this protein was also polysilated, as was this protein, but they didn't realize it at the time because this methodology was so poor. And so what we did was actually to develop a new methodology that would allow us to isolate polysilated proteins more robustly. And it's based on this protein endo-N. This is a really cool protein. So it's a polysialic acid binding protein, but it actually comes from the tail spike of a virus that was isolated from sewage in the 60s, I think. And so this protein is normally on the outside of a virus and is found in sewage. The protein is virtually indestructible, and it binds to polysialic acid, and nothing can wash the polysialic acid away. You can treat it with detergent, you can treat it with high salt, you can do whatever you want to it. And so what we did was to engineer this protein to have a his tag for purification and then an Avi tag. And an Avi tag is a short sequence that allows for site-specific biotinylation. What that means is that you can use streptavidin-coated beads to specifically bind to your protein of interest in a near covalent manner. And so that allowed us then to incubate these beads with serum. The lectin, the polysi binding protein, would specifically bind to polysialic acid and whatever protein it was attached to. And then we could wash with really strong detergents that could wash away any non-specific protein. You can't do this with immunoprecipitation. And then we did traditional proteomics to identify the proteins. The first thing that we did this on was just a cell line, a natural killer cell line that we knew made polysilated NCAM, or CD56. It's the most well-studied polysilated protein. And so this is the lysate from those cells, looking at polysialic acid here and NCAM down here. You can see the glycosylated form of NCAM up top and the non-glycosylated down here. In the flow through, a little bit flows through and the non-glycosylated form flows through. There's nothing in the wash. And then the beads, you see lots of polysialic acid and the high molecular weight NCAM. If you pre-treat the lysate with an enzyme that hydrolyzes polysia, you lose all of your polysia signal and you only see NCAM that is lower molecular weight. But importantly, all of it flows through, none of it sticks to the beads. And so then we take these two beads, we send them for proteomic analysis with our collaborator, Nick Scott at the University of Melbourne in Australia. And this is the data we got. So for those of you who aren't used to looking at volcano plots, the further to the right you are, the bigger the difference between this here and this here. And the higher up you are, the more confidence you have. So we were really excited to see NCAM. Instead of that one peptide that you would have got with immunoprecipitation, we actually see 33 unique peptides and 46% sequence coverage. If you've ever done proteomics, you know that this is spectacular for a membrane protein, a highly glycosylated membrane protein. We also found some other proteins down here, including the polysiallotransferase, which is known to autopolysylate, which is exciting, and a couple of other proteins that have actually turned out to be really interesting in other cell types. And so then we wanted to apply this to the breast cancer system, where we had a cell line that we knew was polysilated, but we didn't know what the polysilated protein was. We wanted to do this to show that we could actually find a new polysilated protein with this method. So we did the same thing over here, and we discovered a bunch of proteins, including QSOX2. So one of the things I don't think I mentioned is that there are only like maybe 12 polysilated proteins known. And so with this method, finding even one more protein is just scientifically really interesting. So we found this protein QSOX2, again, 24 unique peptides, 42% sequence coverage. We were really excited about it. QSOX2, when I first discovered this, I guess this was about 8 or 9 years ago when I first identified QSOX2, long before we published it. I looked in the database, the PubMed database, and there are only 7 papers that actually mentioned QSOX2. It was this totally unknown protein. And in the time since, there have been a number of papers published that shows that QSOX2 is very strongly associated with cancer progression and metastasis and poor prognosis in breast, lung, and colorectal cancers. So it was really fortuitous that we found this protein is polysilated. It made complete sense. We went on to validate that it was polysilated using reciprocal co-immunoprecipitation and showed what kind of glycans it was on, but I won't go into too much detail. So the idea was here with the breast cancer that hopefully we could actually look for polysilated QSOX2 in serum to be able to find a biomarker, not to find a biomarker, but to be able to quantify it in serum using that ELISA, so that we could potentially identify who was going to get cancer, breast cancer, and whether or not it was going to progress. The problem is that QSOX2, we are let down by our analytical reagents. We cannot find an antibody to QSOX2 that is specific and sensitive enough to work in serum. You can see in our Western blots, these 2 background proteins complicate things. When you try to do an ELISA with QSOX2, you get really high background no matter what you do. The antibodies, and we've tried every single antibody out there, it just binds non-specifically to serum components. And so we had to abandon that line of work. But we came back to scleroderma. So we use the exact same strategy, same idea. Is polysia dysregulated in the serum of patients with scleroderma? And if so, could we identify the proteins? And if so, could we use them as a biomarker for disease progression? And so the first thing we did was an ELISA just of polysialic acid in scleroderma serum. And you can see that there's an increase in patients with scleroderma. This increase correlates really beautifully with the modified Rodnan skin score. This is a measure of skin thickness. So in patients with high polysia, you also have thicker skin. And so it was a really nice evidence that what we're seeing in serum was reflective of what is happening in the skin. And so this data was published, but we've since gone on to add more samples, and now we've disaggregated by sex. And this is where it got really interesting. So you can see in women in green that the amount of polysia in serum goes up, and it correlates really nicely with disease activity. There's a very clear difference between even limited limited scleroderma and diffuse scleroderma. And so that is extremely exciting. The same is not true for men. So definitely there's an increase, not as significant an increase, but an increase nonetheless in patients with scleroderma. But there is absolutely no difference between the limited and the diffuse forms of scleroderma. So this sort of backed up our idea that whatever sex differences we were seeing in scleroderma, polyscia was actually recapitulating that to some degree. And we were very happy that women, we were seeing this trend in particular, because if you remember, we know that men almost always progress to diffuse scleroderma. We don't need a prognostic marker for men. We need one for women. And so very clear sex differences. This is in early this is very early, within 3 years of diagnosis. And so it is possible that some of these women are actually going to progress to early diffuse within the next year or two. And so one of the things that we really want to find out is, are some of the women up here eventually going to become this particular cohort? So clear sex differences, but this, for me, is super exciting, but not a big enough difference for an actual biomarker. I would like to, we can continue to follow just quantifying polyscia, but I would like to also come at it from another direction, which is to look for the protein. There is already polyscia in healthy serum, comes from the immune system. But if we could find a protein that is specifically in scleroderma serum, then we might have lower background levels that would allow us to increase the sensitivity of our assay. And so that's what we did next, was to identify those proteins that are polysciolated. And so here's the healthy data from serum, the volcano plot that I've shown you before. And we identified in healthy serum that CD56 NCAM protein, again, that was expected, and ST8-SIA4, that polysialotransferase. So that was great. And then we looked at scleroderma serum, and we also saw those two proteins. But now what we're seeing is weird. There's no other statistically significant proteins, so they would be up here somewhere. But we no longer see this really nice symmetrical volcano plot. What we see is that there are a lot of proteins that are very different in the samples that have polyscia, but that they're not reproducible. And so that tells us that there's something going on with our assay. And so if we come back to looking at our assay methodology, the culprit, it turns out, is our polyscia binding protein. So this protein is quite large, and it carries all the way through the analysis. And so what that means is that if you, for example, if you fully saturated this polyscia binding protein with one polysciolated protein, so let's say it's polyscia NCAM, 100% of these are attached to polyscia NCAM, then you would have a 1-to-1 ratio of NCAM and this protein here. And that would show up in the mass spec. But what we're seeing in serum is that there are probably multiple polysciolated proteins. And so that means the signal that comes from the proteins we're interested in are far outweighed by this protein here. And with mass spec, it's just a matter of numbers, especially with the technology we were using at the time. All of the signal was coming from, or 90% of the signal was coming from this protein, not the proteins it was attached to, because there were so many different ones. And so there's new technology that might alleviate that. But at the time, what we had to do was to upgrade our technology to get rid of that protein. And so the way that we did that was new technology. We decided to use an enzyme to enzymatically modify polysialic acid with that biotin. So instead of attaching the lectin to the beads and then using that to bind polyscia, we attached the biotin directly to the polyscia itself. Okay? So that's shown here. We used an enzyme to do that. And so now we do the same thing. We attach to streptavidin agarose. And one of the things that we had to do to actually be able to immunoblot this was an ammonium sulfate precipitation of serum, because albumin is present at such ridiculous concentrations that it complicated any of our immunoblotting. So we removed the albumin. And then what you can see here is that where we've treated the pellet with the modified sugar. So we have the reaction here. Without the modified sugar, you get no signal from the biotin. And then with the modified sugar, you get really nice signal. And you can see all of the different proteins in here. There's lots of really interesting proteins. And then we passed it through the beads and looked at the flow through. We wanted to make sure that there was a little bit left in the flow through so that we knew the beads were saturated as we could get them, but also that there was less signal here than here, so that we knew the material was attached to the streptavidin beads. And we did proteomics. So one of the cool things about this method is that it doesn't rely on really long polyside chains. You can also see shorter ones. And this is what the volcano plot looks like for healthy serum. NCAM is the most abundant protein as expected. So that kind of validated our assay. But now we're starting to see, because our signal to noise ratio is so much better, we're not getting that contamination of the polysialectin. We see so many interesting proteins. There's galectin binding proteins, there's platelet proteins, there's really interesting polysialylated proteins in serum. And so then the question is, what does scleroderma serum look like, and how does it compare to healthy serum? So this is the comparison now of scleroderma on this side to healthy serum on this side. And what you can see is that there are several proteins here that are really, really, really enriched in scleroderma. And this is a cliffhanger. I can't tell you what they are, because we are hoping to follow up on these to actually develop a biomarker. What I can tell you is that they do actually come from the fibroblasts. So our hypothesis that the fibroblasts were secreting proteins that are polysialylated was correct. And we do not see these proteins at all in healthy serum. And so the thing to do now is to follow up on all of this data. We've set a really beautiful stage for looking at developing this biomarker to predict disease trajectory in women. So we have the original polysia ELISA, where we're waiting. We've collected the serum at initial time of diagnosis and at 1 year follow-up. Now we're just waiting to 3 years to see how the disease progresses in these patients, to see if those who we thought maybe had limited scleroderma actually end up with diffuse scleroderma. We take those two proteins that I just showed you, develop the ELISA, and quantify them in the sera of patients who developed either LLC or diffuse scleroderma. We now have serum from very early patients, and we're starting to collect follow-up data. So now we're able to do this. It's not like cancer, where there are biobanks available because there's so many people who have been collecting serum for so long. Scleroderma is way under-resourced and understudied in comparison. So we actually need to collect the samples and then wait to see what happens to these patients. The other thing is that the scleroderma serum that I showed you, those proteomics results are from a patient with diffuse scleroderma. And so we wanted to do it in a patient we knew had the most polysialic acid, but that's not to say that patients with earlier forms of the disease don't have different kinds of dysregulation. And so we want to re-look at proteomics from serum samples from patients with pre-scleroderma and with very, very early scleroderma so that we can see if there are different proteins. We're looking to see if there's a correlation, particularly in the immune systems, going back to that T cell data I showed you and the monocyte data. Can the immune system predict response to stem cell transplantation? And then lastly, is there potential for polyscia in fibroblasts? If we can inhibit it, is that a therapeutic target? And we have some really compelling data that shows that the glycan may actually be important for inhibiting apoptosis and things like that. So it is definitely a survival mechanism for these fibrotic cells, and could potentially, if we disrupt it, it's very, very early stages here, but could potentially be a novel therapeutic target. So with that, I just want to, again, thank the people who did the work, thank the people who have funded the work. Thank you for your attention, and happy to take any questions.
Thank you, Lisa. So let's jump into the audience questions. So what other fibrotic diseases might involve the dysregulation of polysialic acid?
That's a great question. Certainly there's a fibrotic component to cancer. And so we don't have Louise's fantastic technology, but we did get a spatial multiplex microscope that will allow us to start looking at whether or not polyscia is associated with fibrotic regions in two-dimensional space. And we're looking at that in scleroderma and in cancer. We're not really interested in looking to see if serum polyscia is associated with more fibrotic cancers. I think that that's probably not super useful. Yeah. So
Sure.
definitely cancer.
Okay. And is it related to minimized tryptophan or cysteine? Sorry.
Is it related to? So no. I'm not quite sure of the context of the question. The glycan itself is on asparagine and serine or threonine. Okay. So those are important.
Great. And I think that's it. So thank you, Lisa. If anyone has any further questions, please consider reaching out to the speaker directly. Her contact is shown on the screen. So thank you all to the whole panel for sharing your work. We really appreciate your presence here today. I'd also like to take this opportunity to once again thank our symposium sponsors, Fuji-Rabio, Lyco Microsystems, Selecta, Sampled, Ampersand Biosciences, Biorad, and Aviva. So as a reminder to the audience, the symposium will be archived on the scientist website, and you'll receive an email notifying you when the recording is available. Make sure to follow the scientists on social media for updates on future events. I'd like to thank everyone who took the time to join us today, and particularly those of you who shared your questions and comments. On behalf of the scientists, I'd also like to thank our panel of speakers, Jorge Moscat, Katie Irwin, Francesca DeSimone, Louise Alvarez, and Lisa Willis, as well as our sponsors, Fuji-Rabio, Leica Microsystems, Selecta, Sampled, Ampersand Biosciences, Biorad, and Aviva. Thanks, everyone, and goodbye.